K Biology, Unit 2 Speedrun. Let's go. First, a note on ATP and ADP. ATP, or adenosine triphosphate, functions as the intracellular energy currency in all organisms. It contains a nitrogenous base, or adenine, a ribose sugar, and 3,1,2,3 inorganic phosphate groups. When the bonds between these groups are broken, energy is released. ADP, on the other hand, only has 2 inorganic phosphate groups. It only have 2 of these. If another phosphate group is added, it will become ATP. Adding a phosphate to something is called phosphorylation. Basics here about photosynthesis. So photosynthesis is a process where producers take in inorganic molecules and produce organic substances such as glucose. This will occur in chloroplasts, which contain the green pigment chlorophyll. There's a light-dependent stage which involves photosystems and light-independent stage which is also called the Krebs Cycle. Carbon dioxide and water are the main reactants of photosynthesis. Glucose is the main product and oxygen is the byproduct that is released. So within the chloroplast, the light-dependent reaction occurs in the thylakoid membranes. A stack of these is called a granum. It kind of looks like a stack of green pancakes. The light-independent stage, or the Krebs Cycle, occurs in the stroma, which is like the background material of the chloroplast. Here we have the structure of a dicot leaf. So on top here we have the upper epidermis, and this is a transparent layer of cells. It's transparent because it doesn't have any chloroplast. It is coated with a watertight waxy cuticle, basically to limit water loss. Down here we have the lower epidermis. This is a thin layer of cells, and this includes guard cells and stomata. Basically this would allow diffusion and transpiration of gases such as oxygen, carbon dioxide, and water vapor. Spongy mesophyll cells, which are above here, this is a loosely, irregularly packed layer of cells with lots of air spaces. This would facilitate gaseous exchange between the palisade mesophyll, which is here, and the stomata here. In the box in green, these are the palisade mesophyll cells. This is where the party going on. This is where most of the photosynthesis is taking place. As a result, this would contain a very large number of chloroplasts for light absorption. Not only that, but there's a very large vacuole, and what this would do is that it would push the chloroplasts onto the outer edges of the cells. This would be to maximize light exposure. Most of the sunlight would be propagating through the upper epidermis, which is transparent. So the palisade mesophyll cells are adjacent to vascular bundles, which you can see here in blue. This contains the xylem, the phloem, and the cambium. The xylem supplies water to the spongy mesophyll cells, which will then flow to the palisade mesophyll. Now, the palisade mesophyll also has very thin walls for rapid diffusion of gases. Remember, it needs carbon dioxide for photosynthesis. Photophosphorylation, that's a big word, so let's break the word down. Photo means use of light. Phosphorylation is the addition of a phosphate to a compound. So the concept is thus simple. Photophosphorylation is a process that uses light to attach a phosphate group to an organic compound. So remember, ADP has 2 phosphates, ATP has 3. So the phosphate is added to ADP using energy from sunlight to turn it into ATP. So photophosphorylation can be cyclic, and this is usually in bacteria, or non-cyclic, which is in green plants. This involves photosystems. Photosystems are chloroplast protein complexes, and there are 2 photosystems, photosystem 1 and photosystem 2. Photons, or light particles, are caught by photosystem antennae complexes, which usually are like glycoproteins on top here. And what this would do is that it would stimulate the movement of electrons, kind of like little balls in a pinball machine, where we go pling, 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 pling to all these complexes here. So these electrons move through a chain of electron carriers, eventually driving the formation of ATP from ADP, and this would occur through an enzyme called ATP synthase. So in basic terms, this is what's happening here. Light is absorbed by photosystems. This would stimulate the movement of electrons. These electrons bounce from carrier to carrier. Eventually now what would happen here is that this would drive hydrogen ions from on top here to go through these protein channels, and this would be used to power ATP synthase to phosphorylate ADP to turn it into ATP. This occurs in the thylakoid membranes. So things are going to get a little more complex now because we have the inclusion of coenzymes, NAD and NADP. These are reducing agents. NAD is used for respiration, NADP for photosynthesis. Think the P at the end for photosynthesis if you tend to mix them up. Easy way to think of them as catcher's mitts. Think of them as little gloves that will open up and they will catch hydrogen ions and any associated electrons. So whenever NAD and NADP catch a hydrogen ion and 2 electrons, they turn into their reduced forms NADH and NADPH. Think of the H here, NAD plus the H is NADH, NADP plus the H is NADPH. This is also called reduced NAD and reduced NADP, same thing. So what can happen now is that think of these now as the gloves holding the ball, and they can toss out that ball, or think of them as charged batteries that can release energy. So NADH and NADPH release the hydrogen ion and the 2 electrons to once again become NAD and NADP. These ions and electrons now, these can be used to stimulate certain reactions. Here we can see a visual of it. NADPH, it is a reduced form because it has the hydrogen ion and the electrons here. So imagine like it has this little ball of energy. When it oxidizes and turns back into NADP, it releases that hydrogen ion and the electrons. So once again, think of the reduced form as it having built up energy potentially waiting to be released. So remember I said there's 2 types of photophosphorylation, cyclic and non-cyclic. So in this case here, cyclic photophosphorylation mainly occurs in bacteria. This is the synthesis of ATP during the light-dependent stage. And only one photosystem is involved here, not both of them. So what happens here is that light would stimulate electrons in photosystem 1. Only one photosystem is involved here, not both of them. And when these electrons get excited, they're going to move along this electron transport chain. These are made up of electron carriers, which you could think of these as little protein complexes that are able to shuffle electrons along. Think of them like a conveyor belt for electrons. So this is going to continuously happen, but this is going to happen in a cyclic manner because the electrons that get released actually return to the photosystem, so you could think of them as being continuously reused, at least until they run out of energy. So these excited electrons, this would drive the formation of ATP. Non-cyclic photophosphorylation is a lot more complicated. It involves both photosystems, photosystem 1 and photosystem 2. This mainly occurs in green plants, and this also involves the coenzyme NADP, which turns into NADPH when reduced. This also involves the photolysis or splitting of water molecules using light. So first of all, what happens here is that light energy or photons gets absorbed by photosystem 2. This would stimulate an electron to move like this, pling, 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 pling. So it would move from photosystem 2 to photosystem 1. The light would also be used to split these water molecules into 3 things: hydrogen ions, electrons, and oxygen, which would leave through the stomata of the leaf. These electrons would continue the process, pling, 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 pling. Eventually now the electrons would be accepted by the NADP and would reduce it to form NADPH. So this would be electrons and hydrogen ions. At the same time, the electrons would have provided energy to pump any hydrogen ions that went up here, down here through these protein channels. So down here you would have a buildup of hydrogen ions. These would then flow through this enzyme called ATP synthase. This is basically attached to our protein carrier here. This comes like fuel for ATP synthase. It would turn ADP into ATP, basically it would phosphorylate ADP to turn into ATP, hence the term photophosphorylation. So this is non-cyclic because the electrons at the end do not return to photosystem 2. The additional electrons are actually provided through the splitting of water molecules. Here we have a table showing the differences between cyclic and non-cyclic photophosphorylation. Feel free to pause the video to review them. So here we have the Calvin cycle. It looks quite complex, but remember the main goal here is to produce glucose, which is a 6-carbon sugar. But not just to produce glucose, but also to ensure that the cycle keeps continuing. So first of all, we're going to start with this 5-carbon sugar called RUBP. This is called ribulose biphosphate. This here would have to add a carbon to it. This carbon comes from carbon dioxide. Rubisco is an enzyme that would catalyze this process, the addition of the carbon from carbon dioxide to the RUBP molecule here. So 5 carbons plus 1 carbon will give you a 6-carbon sugar. This is not glucose yet though, this is just an intermediate. So this 6-carbon intermediate would have to now break down into 2 3-carbon compounds, which is what you're seeing here. Each of these is called phosphoglyceric acid or PGA. Now remember all of the ATP and NADPH that was built up previously? What would happen now is that the energy from those, they sort of built up, and remember I said those are like charged batteries. What would happen now is that those would be reduced, the NADPH would be reduced from NADP, and ATP would be broken down from ATP, and this energy here would be used to turn PGA into a molecule called triose phosphate or G3P. This G3P is a very important molecule because this is actually what would go on to form glucose. But this is not the end of the cycle because we have to ensure that the cycle keeps continuing. So we need to regenerate REBP. So not all of the triose phosphate molecules are used. So any excess triose phosphate molecules are recycled, and these now will be used to regenerate REBP using ATP. Then the cycle will restart. So basically, in a nutshell, this is what happens here. Rubisco would attach the carbon from the carbon dioxide to the 5-carbon compound REBP. So this 5 carbon plus this 1 carbon would form a 6-carbon intermediate. This 6-carbon sugar now breaks down into 2 PGA molecules. These are 3 carbons each. Using ATP and NADPH, using the energy from those, this PGA would convert into a triose phosphate. This is the precursor to glucose. This would go on to become glucose. Not all of these are used because some of them must be recycled in order to regenerate REBP. That is the Calvin cycle. So now to talk about limiting factors. Well, first of all, photosynthesis requires the following things. You need energy from sunlight, carbon dioxide and water, a reasonable temperature, usually 25 to 35 Celsius, and chlorophyll. So when one of these factors becomes reduced, so will the rate of photosynthesis. So for example, photosynthesis will not occur if you don't have water. It will not occur if you don't have sunlight. It will not occur if you don't have carbon dioxide. So on the graph on the right, we could see that the rate of photosynthesis on the y-axis and the light intensity on the x-axis. Basically, where the line is going up before it plateaus, this is where light is a limiting factor. Basically, it means that if you was to drop the light intensity at any point, the rate of photosynthesis would decrease. This means that light is actually limiting the rate of photosynthesis at this point. So a limiting factor is one that when increased or decreased influences the rate of a reaction. Now at a point here, we can increase the light intensity and it doesn't influence the rate of photosynthesis. This is because it has reached a saturation point. There's only so many electrons that can be stimulated in the chlorophyll molecules. So at the point here, we have already stimulated all the electrons in the chlorophyll molecules. So another factor has become limiting. In this case, here's the number of chlorophyll molecules. If you was to add more chlorophyll, the rate of photosynthesis will go up. But light at this point is no longer a limiting factor. Carbon dioxide is another limiting factor. So for example, in low levels of carbon dioxide, we have a lower rate of photosynthesis than if we have a higher level of carbon dioxide. So this is because carbon dioxide is of course required to donate carbon atoms to ribulose biphosphate or REBP to produce 6-carbon sugars. And if the carbon dioxide concentration is limited, the Calvin cycle process is slowed down and photosynthesis decreases. So at high levels, it reaches a saturation point where all of the Rubisco enzymes are working at optimum efficiency. So since Rubisco is an enzyme, and we know some about enzymes, and enzymes have active sites which fit substrates, and they can be warped or permanently altered or denatured due to heat. If you was to increase the temperature too much, its efficiency would be affected. So too much heat can denature the enzyme, and that would cause the photosynthetic rate to drop to 0. So there are many ways you can apply this knowledge, and this would be through greenhouses. Artificial light levels can be adjusted to maximize photosynthesis, or with growth chambers, where temperature and carbon dioxide levels can be regulated for sensitive crops such as groundnuts. So it's important to know at what points factors become limiting and not limiting. Alright, a quick reminder on ATP and ADP. Remember, ATP is like a charged battery. It has 3 inorganic phosphate groups. When the bonds between these groups are broken, energy is released, and it turns into ADP. ADP only has 2 inorganic phosphate groups. If another phosphate group is added, it becomes ATP. And remember, adding a phosphate is called phosphorylation. And a quick reminder about NAD and NADH. So remember, these are coenzymes, and NAD can accept hydrogen ions and become NADH. This is reduced form. So if NAD was to catch a hydrogen ion and 2 electrons, it becomes NADH. So as NADH, it can release those hydrogen ions and associated electrons, basically during oxidation. So there's another coenzyme, FAD, which is quite similar, but except it can accept 2 hydrogen ions instead of 1, and it becomes FADH2. Alright, so let's come to understand what glycolysis is. So glycolysis translates to the breaking apart of glucose. Energy is released as a result. So this is step 1. You're looking at the blue rectangle here. Glucose has 2 phosphate groups attached to it, meaning that it is twice phosphorylated. Here we have the glucose, and here we have it turning into a compound called fructose biphosphate. The two phosphate groups come from these two ATPs here. So two ATPs have each donated a phosphate group and become ADPs. So this is the glucose, these are the two phosphate groups here. So the glucose first becomes glucose 6-phosphate. This isomerizes, so basically rearranges the structure into fructose 6-phosphate. That's after it gets the first phosphate. And after it gets the second phosphate, it becomes fructose biphosphate. That is step 1. Step 2. This fructose biphosphate that was found, this is unstable. So what happened here is that this would split and become two 3-carbon sugar molecules. This is called G3P or glyceraldehyde 3-phosphate. Sounds familiar? So these must now have their phosphate groups removed to become a 3-carbon sugar called pyruvate. Pyruvate molecules are what these glucose molecules have to break down into in the end. Pyruvate is our end goal. Step 3. A dehydrogenase or hydrogen removing enzyme is needed to convert the G3P to pyruvate. So dehydrogenase would only work, it would only remove that hydrogen if NAD is present. Remember, NAD is like a catcher's mitt that would catch that hydrogen ion that is being removed. So dehydrogenase would remove the hydrogen ion, NAD would catch it and turn into NADH. Now, two ADPs are going to become phosphorylated to become two ATPs. So we remove these phosphate groups here. And now we've turned this G3P into pyruvate. Now remember, this part here occurs twice because we have two G3P molecules. So you're going to get two molecules of pyruvate in the end. So key things to remember. 2 ATP actually needed to start the process to convert this glucose into fructose biphosphate. Without these 2 ATP, this process would never start. 4 ATP are made in total in the end, which you can see here, 1, 2, 3, 4. But since 2 ATP were needed to actually start the process, only 2 net ATP are gained from the process of glycolysis. So this is called substrate level phosphorylation. We also gained 2 NADHs from reducing NAD. Here we have the mitochondrion. The pyruvate formed from glycolysis would enter the mitochondrion, and much of the action occurs in the mitochondrial matrix, which is the inner part, the fluid-filled space inside. ATP is made here due to the energy from the flow of hydrogen ions. The inner membrane, which you can see that there are little folds here called cristae, contains the ATP synthase enzyme, which synthesizes ATP from ADP. It would also contain the electron transport chain. So the cristae are inward folds of the inner membrane, and the more cristae that are present is the more ATP synthase. And thus the more active is the mitochondria and the more energy could be released. Here we have the 4 stages of aerobic respiration. This is going to be a lot to take in, so feel free to pause the video and read everything first. But we're going to go through it step by step as we go along. First, there's glycolysis. That is what we just looked at. That's going to turn the 6-carbon glucose into 3-carbon pyruvate molecules. The link reaction is going to turn these 3-carbon pyruvate molecules into a 2-carbon acetyl coenzyme A. Where did the extra carbon go? Where it left as carbon dioxide. This is the carbon dioxide that you're going to breathe out. This acetyl coenzyme A, this is going to act as fuel for something called the Krebs cycle. And basically what's going to happen here is that there's a 4-carbon compound called oxaloacetate. This would combine with the 2-carbon acetyl coenzyme A to form a 6-carbon compound called citrate or citric acid. So this is also called the citric acid cycle. This was going to build up multiple reduced NADs and FADs, or NADHs and FADH2. Remember, these are like charged batteries. These are like catcher's mitts that are holding balls of hydrogen and electrons ready to be released. Lastly, we have oxidative phosphorylation. All these NADHs that have energy and electrons and hydrogens in them, they're going to oxidize and become NADs. This is going to release energy to drive ATP production along the electron transport chain. The diagram here shows the link reaction. The link reaction connects the glycolysis step to the Krebs cycle, hence the term link reaction. So pyruvate, which has 3 carbons, loses a carbon due to the removal of carbon dioxide. That's what you're seeing here. So 3 loses 1, becomes 2. So it becomes a 2-carbon compound. NAD accepts hydrogen to become NADH. And there's an enzyme called coenzyme A, which would convert this 2-carbon compound to acetyl coenzyme A. This is going to act as a fuel. This is going to power the Krebs cycle. Here we have the Krebs cycle. It could look pretty intimidating. Feel free to pause the video just to take a look at it before I explain. Right, so let's start here with citrate. So how is the citrate formed? Citrate, as you can see, is 1, 2, 3, 4, 5, 6 carbons. Remember, acetyl coenzyme A is 2 carbons. So we need a 4-carbon compound to attach to this. That's going to be a compound called oxaloacetate. This is 4 carbons. So this 4 and this 2 would make up this 6 here, citrate. And citrate is what's going to kickstart the whole thing. In the Krebs cycle here, ATP is produced once every cycle. A glucose molecule can produce a net amount of 32 ATP. So acetyl coenzyme A is going to combine with oxaloacetate to become citrate. 2 carbon dioxide molecules are going to leave. So this 6-carbon compound is going to lose 2 carbons. So first it's going to become a 5-carbon molecule, then a 4-carbon molecule. As this happens, hydrogens are being released, and these would be taken up by NADs to become NADH. Here you can see it happening once, twice, and a 3rd time. The exception is here where we are getting 2 hydrogens being released, and this here would be taken up by FAD to turn into FADH2. Now remember, the buildup of all these NADHs and this FADH2 are like charging up batteries, or it's like a bunch of catcher's mitts holding energy in it. So as I said, 1 ATP is formed per cycle, and 32 net ATP is made from 1 single glucose molecule. So the cycle is going to restart as oxaloacetate binds with more acetyl coenzyme A to become more citrate. So if the Krebs cycle was a car, acetyl coenzyme A is the gasoline. You end up with 3 NADHs and 1 FADH2. Later on, these will be reoxidized, and this will drive the formation of ATP. Lastly, we have oxidative phosphorylation. This is the process that uses oxygen to attach a phosphate group to ADP to turn it into ATP. This is facilitated through a series of proteins called an electron transport chain. Electrons are shuttled along the chain and they release energy as they do. Now at the end of the electron transport chain is oxygen, an oxygen molecule. Without oxygen, the circuit is broken and no ATP is made. I like to think of it as a series circuit where the switch is the oxygen molecule, and any loads here like the electron carriers, of course we have electrons passing through the wires here. The battery is going to be all those reduced NADs and FADs that we would have built up from the Krebs cycle. So if the oxygen molecule is not here, none of this could occur. So on this diagram here, which again could look pretty complicated, we're going to break down what's going on into 3 steps. There's step 1, step 2, and step 3. So you're looking here first at step 1. All these reduced NADs and FADs, these are going to release hydrogens, and they're going to become NAD and FAD again. Remember, this is when they get reoxidized. These hydrogens are going to split into hydrogen ions or protons and electrons. These electrons are what you're seeing here. These electrons are going to be shuttled from carrier to carrier along the electron transport chain. They're going to go boop, boop, boop. And at the end of the electron transport chain, there's going to be an oxygen. This oxygen now is going to combine with hydrogen ions, and think hydrogen and oxygen, what's that going to form? That's going to form water. Step 2. The energy from the electrons is used to pump hydrogen ions across channels. This is going to end up here in this intermembrane space. So basically, as these electrons flow along the chain, this is going to encourage all these hydrogen ions to shoot up into this intermembrane space here through these protein channels. So this would allow the intermembrane space to have a more positive charge than the matrix. Up here, you'd have a more positive charge than down here. This would build up what is called an electrochemical gradient. So through a process called chemiosmosis, these hydrogen ions are going to flow from a higher concentration up here to a lower concentration down here, back into the matrix. So this diffusion of hydrogen ions, remember all of these contain energy. This would occur through ATP synthesis in the inner membranes. So remember, as hydrogen ions power the ATP synthesis or ATPases, this would phosphorylate ADP and turn it into ATP, and that is how energy is made. Now, anaerobic respiration is the release of energy from ATP without the use of oxygen. So without oxygen, the electron transport chain cannot be completed. This means that the link reaction, the Krebs cycle, and oxidative phosphorylation cannot occur. Basically, the only thing that could occur is glycolysis. So anaerobic respiration takes another metabolic pathway after glycolysis occurs. Here you can see that here. So from the diagram, as you see in here, glucose is turning into 2 pyruvate molecules, and 2 NADs are being reduced to form NADH. These 2 pyruvates are going to convert into 2 lactates. Think of that as like lactic acid. So this is now this NAD and NADHs. This now is gonna reduce and oxidize and reduce and oxidize, kind of like a cycle, and this is gonna allow glycolysis to occur continuously. This would only produce 2 ATP per glucose as opposed to 32 ATP during aerobic respiration. So much less energy is produced through anaerobic respiration than aerobic, because only substrate level phosphorylation can happen during anaerobic respiration. So in yeast, the process is quite similar, but you're going to see instead of pyruvate converted into lactate or lactic acid, pyruvate is going to release carbon dioxide molecules to form ethanol. And ethanol is then going to convert to ethanol. Ethanol is a major constituent of what we know as commercial alcohol, things that you'd find in beer and rum and wine. The carbon dioxide has been released through this reaction in the yeast. This is used to raise bread. Here we have a table comparing aerobic and anaerobic respiration. Feel free to pause the video and go through it yourself. Here we have the mitochondrion again. Note the annotations on the diagram. Also note the materials that are going into the mitochondria, which are NADH, ADP and an inorganic phosphate, pyruvate, and oxygen. And also note the materials that come out of the mitochondria during respiration, which is NAD, which is the oxidized form of NADH, ATP, carbon dioxide, and water. Feel free to pause the video and read the annotations. Let's look at energy transfer. A food chain depicts the feeding relationships in a given ecosystem. It is comprised of various trophic levels. In a food web, organisms may feed from various trophic levels. In this particular terrestrial food chain, we can say that the grass is the producer or the autotroph. What the grass can do is that it can transform the electromagnetic energy from the sun into stored chemical energy, in this case here sugars. This grass is consumed by the grasshopper. The grasshopper is consumed by the bluebird, then the snake, then the owl. Each of these is called a consumer because it feeds on the organism previous to it. So these would be assigning names in order: primary, secondary, tertiary, and quaternary consumer. The one at the top is also called an apex predator. All of these organisms must die, of course, and decomposers would break down the organic matter in these cells into inorganic molecules such as nitrogen and carbon, and they would return it to the atmosphere or the soil. Decomposers are not considered consumers. These usually are fungi or bacteria. So the transfer of energy in a food chain is known as an energy flow, not a cycle, meaning that it cannot be returned to its source, in this case here the sun. An average of 10% of energy is transferred from one successive trophic level to another. So for example here, let's say we have the energy from the sun is taken up into these trees and this grass. These producers, let's say a particular producer has 10,000 kilocalories. The rabbit consumes the grass, and this only gets 10% of the amount, which would be 1,000. The snake consumes the rabbit, gets 10% of that, which is 100, and the eagle only gets 10% of that, which is 10. So where did the other 90% go? 90% is lost to the atmosphere as heat from when metabolic life processes have to be performed. This would be Grimmner, growth, respiration, irritability, movement, nutrition, excretion, and reproduction. All of these take energy, and when they use energy, the heat is lost to the atmosphere. So organisms that are high up in the food chain, for example hawks or the eagles in this case, receive very little energy from feeding on trophic levels directly below them. For example, when the eagle eats the snake, it only gets 10 kilocalories. But if the eagle was to feed on the rabbit, it would get much more. So in this case here, they may have to feed on multiple lower trophic levels to have enough energy to sustain their biomass. So when we talk about energy losses, we have to keep in mind the law of conservation of energy, which says that energy cannot be created nor destroyed, but can only be converted into different forms. In this case here, as previously mentioned, organisms must respire and perform life processes to survive. So the 10% of energy is stored in the cells, available for transfer if the organism is eaten. So for example here, let's say that this plant material that's eaten by this caterpillar is 200 joules. The caterpillar would only have 20 joules stored in the cells. This is because 80 joules would be used for cellular respiration, and 100 joules might be excreted or ingested. So only 10% is actually stored and 90% is lost to the atmosphere. Now we also have to remember that not all parts of the organism may be consumed during feeding. So for example, when carnivores eat another animal, they may not digest the bone material and the cartilage, and they may not feed on it at all. So the energy in that is sort of lost. Lastly, not all molecules consumed would be digested and assimilated. For example, when we humans eat vegetables, we don't have the enzyme that breaks down cellulose. So all that cellulose is just used as fiber and roughage to move food along the alimentary canal, is not actually assimilated into the cells. In terms of photosynthetic losses, well, the photosynthesis can be quite inefficient. In fact, most of the energy from sunlight is actually not used for photosynthesis. This is because, well, one, sunlight is often reflected from leaf surfaces. This could be through the shininess of the waxy cuticle or just the color of the leaves themselves, where the chlorophyll is just inaccessible. Sunlight may enter the leaf but miss the chlorophyll molecules altogether. So even though the palisade mesophyll cells may be packed very tightly with the chloroplast on the outside, not all of them may be able to absorb that sunlight. Thirdly, only certain ranges of light wavelength can be absorbed by that chlorophyll. The process of photosynthesis itself uses energy. In fact, if you were to take the gross productivity of the plant, you would have to subtract the energy used for cellular work to actually get the net productivity. Lastly, sunlight may not even come into contact with the leaves. The leaves may be shaded, or the leaves may be inaccessible by sunlight. It could even be the anatomy of the leaves. The leaves may not be broad enough. The leaves may be curled. The petiole may not have oriented the leaves towards the sunlight. All of this would be considered photosynthetic losses. Ecological pyramids. So pyramids help visually conceptualize data in ecosystems according to trophic levels. So there are three main types: the pyramid of numbers, pyramid of biomass, and pyramid of energy. So there are several limitations when dealing with this. First thing is that ecosystems are never static. Population numbers and dynamics are always changing. Organisms would have to be observed for a very long time in their habitats. So for example, it's very, very difficult to get a true count, a true physical number of organisms in an ecosystem, because they're always reproducing and they're always dying. It's never static. Secondly, organisms can occupy more than one trophic level simultaneously. So for example, a bluebird may feed on berries, and that would make it a primary consumer, but it may also feed on caterpillars, which would make it a secondary consumer. Lastly, feeding relationships may change as seasons pass. As organisms migrate during winter, or certain foods become inaccessible or may mature at different periods of time, this there would affect feeding relationships. Here we have a pyramid of numbers. The pyramid of numbers represents the number of organisms that fall within a particular trophic level. So for example, here we have 12,000 producers, 3,000 primary consumers, 120 secondary, and 3 tertiary. You'll see that typically the number will decrease with each trophic level, as there are usually less predators than prey. So for example, here let's see these primary consumers are rabbits, and this secondary one is snakes. You won't have more snakes than rabbits because the snakes would run out of food over time. Now, inverted pyramids can occur if the producer is a single tree, for example, and parasites are involved. You would have multiple parasites feeding on one particular organism. A pyramid of biomass accounts for the total mass of a population or trophic level occupying a certain area of the environment. So for example, a single tree would have a greater biomass than all the termites or birds living within it. Biomass is measured in grams per square meter. Pyramid of energy represents the flow of available energy from one trophic level to another. Sometimes this is called a pyramid of productivity. This would always have a typical pyramid shape, because the amount of energy in the producers would always be more than the amount of energy through each successive consumer. So lower trophic levels would have more energy available in their cells than higher levels. Let's take a look at the nitrogen cycle. Now, 78% of the atmosphere is nitrogen. Now, most of that nitrogen is not really usable by organisms such as green plants. That nitrogen gas must be converted into materials called nitrates. Nitrates are taken up by plants to make amino acids. So first, in order to do this, nitrogen fixation must occur. Nitrogen fixing bacteria in the soil or living in the root nodules convert this nitrogen gas into a compound called ammonium. Now, this can also be done by lightning or synthetically by the Haber process. Next, we have nitrification. This ammonium is going to be converted into nitrites and then into nitrates by nitrifying bacteria. Remember, these nitrates can then be taken up by plants. This is called assimilation, and they can then be used to form amino acids. Animals can then eat plants and get the nitrogenous compounds into their cells. In that way, nitrogen is passed along the food chain. Lastly, we have denitrification. The leftover nitrates in the soil are going to be converted back into atmospheric nitrogen by denitrifying bacteria. And in another step to produce ammonia, we have ammonification. When plants and animals die, or when they release nitrogenous waste, so for example, when animals urinate, they're going to release urea. Decomposers are going to break this material down. It's going to convert this organic material back into ammonia. Energy flow. So we have nutrient cycles, for example, the carbon cycle, the nitrogen cycle, the water cycle. All of these are driven by energy flow. Now remember, energy flows from the sun to producers, which are eaten by consumers. When producers and consumers die, decomposers allow the nutrients from the consumer cells to enter the inorganic nutrient pool. We can see from this flow chart here. The producers absorb energy from the sun. Consumers, such as herbivores and carnivores, eat the producers. Decomposers break these consumers down as well as producers when they die, and they return nutrients into the soil, which is then used again by producers. Now at each step, heat is transferred out to the atmosphere due to respiration and excretion. Now one key thing to remember is that none of this energy can ever return to the sun, thus the term flow and not cycle. Biotic factors consist of members of living populations and their interactions with each other. Examples would include predation, where one organism hunts and eats another. This helps to control several populations in the food web. Competition, you can have interspecies or intraspecies competition where multiple organisms occupy similar niches and must vie for limited resources or territory. Parasitism, one organism benefits while harming the other one. Example would be ticks and dogs. Commensalism, one would benefit while the other is unaffected. Example would be the remora fish hitching a ride on a shark. You would have mutualism, where both organisms would benefit. So this would be like rhizobium bacteria living in the nodules of pea plants, or like oxpeckers and zebras. Lastly, we have altruism, where organisms would cooperate for a common goal. This would be, for example, like ant colonies. Next, we have abiotic factors, which are non-living factors. So we have light intensity, which will of course impact the rate of photosynthesis. We have temperature, which could impact water supply or the type of vegetation. Gas concentrations, which could impact rate of photosynthesis and respiration. pH, which could impact the type of habitat and the water available for drinking. Soil, which include the aeration of the soil, any habitats, the pH, the particle size, the mineral concentrations. Basically, it could impact vegetative growth. We have water, which includes the availability of water, the habitat, the turbidity, that's like the muddiness, the salinity or the saltiness, and the pH. Wave action, which can result in erosion and help moving nutrients and gases around. Humidity with decreased transpiration. Wind speed would increase transpiration. And topography, which is like the layout of the land, would include the different types of altitudes that animals and vegetation can live. Biodiversity is an amalgamation of three factors: species diversity, genetic diversity, and ecosystem diversity. Biodiversity is said to be linked with ecosystem stability. Ecosystem stability is the ecosystem's resilience and ability to return to a normal state after being negatively impacted. Let's say, for example, you have a flood or a hurricane and the ecosystem must be devastated in some way. If the ecosystem was stable before, it would return to its usual state in a short amount of time. So species diversity refers to the variety of different species found within a biome. It does not include the abundance of species or the size of the populations, just the different fact that there are many different species found there. So for example, the savanna here may have many different species of grasses, shrubs, trees, birds, insects, large mammals, and so on. Tropical regions tend to have high species diversity due to the high rainfall and light intensity, so you have a lot of different trees, a lot of habitats, and a lot of water available for organisms. Genetic diversity refers to the variation of genetic information and phenotypes within the populations of the species. So for example, having selective pressures and high rates of outbreeding would increase the size of the gene pool and the number of favorable alleles and traits. So if you remember from Unit 1, the Galapagos Islands had these finches that Darwin studied. And because of their different beak sizes, they didn't have to compete with each other and they were able to survive based on that. So members of the species are more likely to resist disease and adapt to environmental changes, thus raising chances of survival. Ecosystem diversity refers to the variation in ecosystem types in a defined geographical area. An example would be the Aripo Reserve in Trinidad. This includes an open savanna, a palm marsh, and a marsh forest, all adjacent to each other with rivers. So all of this here would encourage intermingling of species and provides many choices of habitats that encourage survival if selective pressures arise. For example, if an invasive species was to come into the open savanna and the organism can survive in a marsh forest, they can easily move there and survive there. So a stable ecosystem is one that has the ability to return to its normal state after a disturbance. So a high species and genetic diversity means that less species are prone to migration or extinction after a disturbance. Some members would have favorable traits to adapt to changes. So let's say, for example, there was a hurricane that affected the topography of a habitat or an ecosystem. If you have enough genetic variation, you would at least have some members that might be able to survive those changes, and those survivors would be able to reproduce and continue to replenish the ecosystem. So high ecosystem diversity as well makes it even less likely that a species will die out, as they can simply adapt to another biome within the ecosystem. So for example, if you have a forest that is beside another type of forest and one forest gets decimated, let's say through deforestation, then the organisms can easily move to the other forest and adapt to that biome. So what is the importance of maintaining biodiversity? So you're going to break this down into different categories: intrinsic, direct, and indirect. Intrinsic refers to the ethical or the existence values. It cannot put a price on it. It doesn't have any known economic use. So one is the ecosystem stability, greater chances of ecological recovery after disturbances. 2, protecting endemic species from extinction. Those are species that are only found in that particular country or area. For example, the Paui in Trinidad is all of a shame when that species goes extinct. The aesthetic value. This is basically how good it does look. Nature itself is a source of relaxation and beauty for visitors. To get rid of that would lead to, well, less places they could post on Instagram. The bequest value. So this is the preservation of the environment so that future generations, which would be like your children and grandchildren, can experience the same thing. Now we have direct values or known economic uses. So one is tourism, of course. Nature tours and trips can earn revenue for the country. People would come to an area just to experience that particular type of nature. Then we have raw materials can be harnessed from the environment, like timber and ores can be responsibly harnessed if reforestation or responsible mining was to take place. Next we have indirect, which means that there's an unknown use or there's a non-immediate economic use. So firstly, our flood prevention. Deforestation and removal of vegetation leaves soils exposed and waterlogged after rainfall. So this can decimate the habitats, but it could also decimate surrounding residential areas. I'm sure we know a few islands that have that problem. Then we have the scientific value. So researchers observe ecosystems and organisms as models for medicinal technology. A good example here is Velcro, which is, that kind of sticky material. This was modeled after birds sticking to clothing, those little things, those little seeds that stick to your socks. In the aspect of genetic engineering, they learned about genes from daffodils that could be spliced into rice to make golden rice for people who are suffering from night blindness, basically to develop vitamin A. So conservation is the discipline of maintaining diversity by protecting species and their habitats. We have in situ and ex situ conservation. So in situ, which basically means on-site conservation, aims to preserve natural habitats and maintain or replenish population numbers. Examples of this would be national parks or wildlife reserves, such as the Aripo Reserve in Trinidad or the Yosemite National Park. Ex situ or off-site conservation aims to preserve target species outside of their natural habitats. Examples of this would be zoos, botanic gardens, seed banks and pollen storage for plants that are in endangered areas, in vitro storage for sperm and embryos, and captive breeding programs, for example for panda bears, so that they can replenish their population numbers. Here I have a table that is comparing the in situ and ex situ types of conservation. Feel free to pause the video and give it a read. Transpiration is the movement of water through a plant, and evaporation through its above ground parts, such as the leaves. Water enters the plant root here is due to osmosis from a higher to a lower water potential up the xylem vessels and into the stems. Water is delivered to the cells in the leaves and excess water is diffused out of the leaves through the stomata. There are two significant pathways in which water travels through the plant cells. This is called the apoplast pathway and the symplast pathway. Let's start with the symplast. So the symplast pathway occurs when water enters the root cells by crossing the plasma membrane. We're looking at the green arrows. And this would enter the cytoplasm and crossing through gaps in the cell wall. These gaps are called plasmodesmata. The apoplast pathway, water does not enter straight into the cytoplasm at first. Instead, it moves cell to cell while staying within the freely permeable cell walls. However, it cannot actually enter into the xylem this way because there are impermeable Casparian strips in the endodermis which will force water to switch to the symplast pathway to reach the xylem. To avoid mixing them up, an easy way to remember which one is which is apoplast. Think A is that it moves A across the cell walls, and symplast, think S, it moves straight through the cytoplasm. Let's look at a cellular diagram of the apoplastic and symplastic routes. The apoplastic route is in the purple, the symplastic is in blue. Now there's one other thing to note here. In the endodermis, which contains the Casparian strips, which will force the apoplastic route to switch to symplast for the water to get to the xylems. Remember, this is where all the water has to end up. The endodermis contains protein carriers for active transport of mineral ions. So as ions are moved into the endodermis, the water potential gradient would steepen. This would increase the rate of osmosis, and this is called root pressure. Basically, there are going to be protein carriers here. This is going to pump mineral ions around the xylem, and this is going to decrease the water potential here in the xylem. This is going to allow more water to flow in, and this is called root pressure. Xylem vessels are used for the upward unidirectional transport of water and dissolved inorganic ions from roots to leaves. They are made of a dead polymer called lignin, used for structural support. There are different structural patterns that this lignin could be placed in, such as spirals and annular lignin, which is like rings. So the xylem has pits in its walls that allow flow of water to and from adjacent cells. Xylem tubes are hollow and continuous. There are no end walls, which could facilitate easy flow of water. So water molecules easily bond with each other. This is called cohesion, but they can also bond to the narrow xylem walls. This is adhesion. This would facilitate capillary action. So transpirational pull occurs when the water molecules exit from the stomata, as you see in here. This will pull the transpiration stream higher and higher as each water molecule exits. Lastly, we have root pressure, and this will build up in the endodermis as inorganic ions are actively transported into the xylem. This would steepen the water potential gradient, allowing more water to come into the xylem. So how do we measure transpiration rates? Well, we use this device here. This is called a potometer. What we do here is that there's a cut chute with an exposed stem and xylem that is securely fitted on one end. An air bubble is introduced into a capillary tube. The starting position is marked next to a ruler. So as transpiration would occur in the chute as the xylem is taking up the water, the air bubble is going to move towards the plant stem. So after a fixed amount of time, the final position of the bubble is marked, let's say for example here, and a distance is measured between its initial and its final positions. So the rate can be determined by measuring an average distance over time over multiple measurements. Next we have the phloem. So the phloem assists with the transport of sucrose from sources to sinks. This is called translocation, whereas the xylem more dealt with water. The phloem consists of elongated sieve tube cells, or sieve tube elements, with few organelles and mitochondria. They are joined to companion cells, which you see in here, and these companion cells have mitochondria to supply ATP for what is called the loading and unloading of sucrose. At ends of sieve tubes are sieve plates, which you can see here, which are perforated discs that will increase pressure and allow mass flow. Here we have a table that compares the xylem and the phloem structure and function. Feel free to pause the video to take note of it. So what exactly is a source and a sink? Well, a source is a photosynthetic organ capable of producing sugars in excess. For example, this would be mature leaves. Sucrose is unloaded from sources into the phloem, so the leaf would deposit sucrose into the phloem. A sink, on the other hand, is a non-photosynthetic organ that does not produce sugars but will require sugars for respiration, for example. Sucrose is loaded into sinks. So for example here, a sink would be roots, fruits, immature leaves, and nectaries. So sugar is transported from sources such as leaves to sinks such as roots. So sucrose would be unloaded from the leaves and loaded into the roots. Now sugars are transported as sucrose because it is less reactive than glucose and is more mobile than starch, so sucrose is kind of like that perfect in between. Let's now get into phloem loading and unloading, basically translocation. So translocation occurs due to hydrostatic pressure differences that occur due to a combination of forces from osmosis and active transport. So let's start here from the leaf cell, where there's a lot of starch broken down into sucrose. So sucrose and other organic ions here, which are assimilates, these are going to be actively loaded onto the phloem from sources. So basically when there's active transport through some protein carriers here, sucrose is going to be co-transported with it. Think of it as opening a little tap and everything is flowing out into the phloem. So because there's more solute here, there's going to be a lower water potential in the phloem at this point, because there's going to be a lot of sucrose here. So because the water potential decreases here in the phloem, water is going to move from a higher water potential in the xylem into the phloem. This water is going to flow from the xylem into the phloem, and the force of this water is going to push all of this sucrose and assimilates downwards here. So as all of these flow down here, the sucrose is going to be unloaded from the phloem into sinks. So for example, here would be a root cell. So as all of this sucrose now gets loaded into this root cell, this is going to increase the water potential in this point. Now because of the pressure of this water, now this is going to push water back into the xylem, where it's going to flow up to the leaves once more to restart the process. So this is a cycle that's going to happen here. So the forces that push the assimilate through the phloem against a concentration gradient are mainly due to transpiration, but active transport as well. Now the main argument against this mass flow hypothesis is that it only accounts for unidirectional flow of assimilates. It doesn't really account for upward flow, only downward flow in this sense. But we know the flow of assimilates is actually bidirectional. The phloem takes substances both up and down. It also does not account for sucrose and amino acids flowing at different rates. Here we have the circulatory system. The human circulatory system comprises the following: the heart, which acts as a pump, blood vessels, which act as pipes, and these involve arteries, veins, capillaries, smaller arteries called arterioles, smaller veins called venules, and blood is the main transport medium. Now only complex organisms with a low surface area to volume ratio would require such a circulatory system. Other ones could just use diffusion, such as amoeba and other unicellular organisms. So blood flows in a unidirectional manner, which means that it never flows backwards. To facilitate this, we have valves. Valves open and close to prevent backflow and synchronize blood flow. If we take a look at the diagram here, we'll see that we have two upper chambers. Each one is called an atrium, and we have two lower chambers. Each one is called a ventricle. This side of the heart is the left side, and this side of the heart is the right side. The left side is oxygenated, the right side is deoxygenated. Easy way to remember it is to remember left oxygenated, right deoxygenated. Here we have the different segments and vessels involved with the heart. We have the superior and inferior vena cava, and these carry deoxygenated blood from the body cells into the heart. Now remember, veins always bring blood into the heart, and arteries always carry blood away from the heart. So here's the aorta, which is an artery, and these carry oxygenated blood away from the heart to the body cells. The pulmonary artery carries deoxygenated blood away from the heart and to the lungs. Pulmonary veins carry reoxygenated blood to the heart and back from the lungs. We have valves, so we have four particular valves you have to take note of. We have these two semilunar valves here, and this is called the pulmonary valve, and this is called the aortic valve. This is because it's connected to the pulmonary artery and the aorta. These are the two valves. This is called the tricuspid valve, and this is the bicuspid valve or the mitral valve. Separating the left and right sides of the heart is the septum, and in particular we have to know the left ventricle. The left ventricle is the muscle dense region of the heart responsible for contracting and pumping blood to the aorta. Systole and diastole. So the heart has two thumps per beat, the second one louder than the other. These two terms refer to systole and diastole. Systole refers to the contraction of the atria or the ventricles, such as when you see up here squeezing or down here squeezing. So this causes atrioventricular blood flow, basically blood flowing from the atrium to the ventricle, or blood flow out of the heart and into the arteries, such as where you're seeing now, into the aorta and the pulmonary artery. Think S in systole as stress or squeezing the blood out. Diastole, on the other hand, is the relaxation of the atria and ventricles, allowing blood to flow into the heart. So blood would refill the chambers at this point. So think D in diastole as the heart de-stressing. There are two electrical nodes in the heart called the sinoatrial node or the SAN and the atrioventricular node or the AVN. So the SAN or the pacemaker, which you see in here, is located in the right atrium. This is the site of the initiation of the cardiac cycle. So the SAN's muscle would contract and produce an electrical impulse, basically what you're seeing here. This would propagate through the atria muscles and cause them to contract. The impulse would then activate another node called the AVN, the atrioventricular node. This is basically between these two atria here, or somewhere along the septum. So what this would do is that this would relay that electrical impulse from the sand to the ventricles and cause those to contract. This process would then repeat. So blood vessels, there are three main types. The arteries, basically what you're seeing here. So arteries are thick-walled vessels with small lumens. The lumens are basically the passageways for blood flow. Arteries transport blood away from the heart to the body cells. Remember, A for artery, A for away. There are thick layers called the tunica media and externa, and these consist of muscle and collagen. Veins, on the other hand, are thinner-walled vessels with larger lumens. These would also have valves, so these would transport blood towards the heart from the body cells. Easy way to remember it is to remember vein in. These transport blood into the heart. If we look at the micrograph here, we can see the artery, which is well defined. We have the tunica media here and the tunica externa, which is the muscle and the collagen. This helps it withstand very high blood pressures. The vein, on the other hand, is easily collapsible, is very thin layers, and the lumen is a lot larger than the arteries. The third type of blood vessel are the capillaries. These are very narrow blood vessels that surround tissues and organs. They form a capillary network. Red blood cells would flow through these in single file. They have a single layer of endothelial cells that facilitate rapid diffusion of things like oxygen and carbon dioxide. But sometimes transport is done through vesicles via exo and endocytosis. Here we can see that the red blood cells flow through them single file, and we can see the endothelial layer here and surrounding body cells. Here we have a table showing differences between arteries and veins. Feel free to pause the video and take a look at them. So if we had to make a plan diagram of the arterial tissue, the histology of it, starting from the innermost layer here, well, first we have the lumen, which is the passageway for the blood. Then we have the tunica intima, which will be highly nucleated. We will see that the nuclei are quite obvious here. Then we have the endothelium, which is a single layer of cells that would separate it from the smooth muscle. Between the endothelium and the smooth muscle, we would have an internal elastic membrane. This could be represented as wavy lines. The smooth muscle and the membrane would make up the tunica media. Finally, we would have another elastic membrane, and the tunica externa would consist of loose fibers and collagen tissue. So let's look at blood cells. We have red blood cells or erythrocytes. This is what you're seeing here. So the function is to bind oxygen to hemoglobin for transport and delivery to cells. The structure is that they have a biconcave shape. Think of a donut without the hole. No major organelles so that they can have more room for hemoglobin, and they are small and flexible so that they can pass through narrow passageways such as the capillaries. Here we have a lymphocyte. So lymphocytes are part of white blood cells. The other term for white blood cells are called leukocytes. So lymphocytes can either make antibodies, and these are called B lymphocytes or B cells. They can send signals. These are called T helper cells, or they can kill infected cells. These are called T killer cells or cytotoxic cells. How they look is that they have large round nuclei, and they have receptors on surfaces to bind antigens. Antigens are detectable foreign substances in the body. This is a neutrophil. So the function of the neutrophil is to engulf pathogens. They are also able to present antigens to lymphocytes. So basically, when they engulf a pathogen, they can inform the lymphocyte that this is what it looks like, and then the lymphocyte can act upon that. The structure of it is that it is a very large cell with multi-lobed nuclei. It is a granulated cell. Then we have macrophages, which are similar in function, but they are less potent. These also used to remove any dead cells. So they have a large, more irregularly shaped, kind of folded nucleus, and they are slightly granulated. Now we have platelets. So platelets are very, very small fragments, and their function is to help form blood clots to slow or stop bleeding as a response to injury. What they would do is that it would activate enzymes and clotting factors to turn fibrinogen in the blood into insoluble fibrin, which would then be used to form a blood clot. So they look like small fragments, and basically they have been broken off from bone marrow cells. Finally, which is not a cell, platelets are not cells by the way, but blood plasma, which is not a cell, this is more the medium. It is a pale yellow fluid, as you can see here, and inside this fluid we would have dissolved nutrients. So this would basically be able to transport soluble proteins, any dissolved materials, nitrogenous waste, hormones, things like that. The cardiac cycle. So the cardiac cycle refers to the sequence of events that comprises one heartbeat. This occurs in 3 stages: atrial systole, ventricular systole, and ventricular diastole. So first, let's start with atrial systole. So remember, the S in systole is kind of like squeeze. So what happens here is that the atria contract, these are the upper chamber, and blood will flow from the atria to the ventricles, the lower chambers. During ventricular systole, the ventricles are going to contract. So imagine that they are going to squeeze, and the atrioventricular valves would close. Blood would push from the ventricles to the aorta and the pulmonary artery. Basically, the blood is being squeezed out of the heart at this point. Then we have ventricular diastole. Remember, the D in diastole is like the heart de-stressing. So both the atria and ventricles are going to relax. Blood is going to flow into the heart from the veins. That's going to be from the vena cava and the pulmonary veins. The semilunar valves are going to be shut to prevent any outflow of blood so that the heart is able to refill with blood. Now let's look at the cardiac cycle from an electrical point of view. First, the sinoatrial node, which is in the right atrium, is going to send out an electrical wave of excitation across the nerves in the atria. This is going to cause the atria to contract. This is basically going to incite atrial systole. What's going to happen now is that that signal is going to propagate to the atrioventricular node, or the AVN. So this is going to pick up the signal, and this is going to send another wave to the ventricles. Now there's actually a short delay in time so that all chambers don't contract all at once between the signal at the SAN and the signal at the AVN. So along the AVN we have Purkinje fibers. These are electrically excitable cells, and these are going to conduct the signal from the atrioventricular node. These are going to move the signal into branches in the septum called bundles of His. So the pathway of depolarization, we'll get more into that later, is going to come down to the ventricles like this, the muscles in the ventricles, and the ventricles are going to contract. This is basically going to be ventricular systole. This is going to contract, and it's going to push the blood through the aorta and through the pulmonary artery, basically squeeze the blood out of the heart. Here we have a graph depicting this cardiac cycle. It looks very complicated, but there's only a few things that we really have to look at. So if you look here at the ventricular pressure, we can see that during ventricular systole, the pressure has an extremely steep increase. This is because the blood is actually pumping through the aorta to the arteries in the body. Because of this, you'll notice that the aortic pressure is actually increasing here as well. This is basically when the blood is being squeezed out of the heart. Now during ventricular diastole, remember, when the heart is de-stressing, is relaxing, you'll notice that all the pressure here has dropped. The aortic pressure has dropped, the atrial pressure has dropped, and the ventricular pressure has dropped, because this is the heart de-stressing, is allowing blood to refill. The actual thump that we hear, the two thumps that we hear with the heart, the lub-dub sound, is actually the first lub is where the atrioventricular valves close, and the dub sound is going to be where the semilunar valves close. Cardiac output refers to the volume of blood pumped out of the heart per minute. This is a combination of the heart rate, which is the number of cycles per minute, and the stroke volume, which is the amount of blood that is pumped out per cycle. The two main factors that affect cardiac output would be adrenaline secretion and the oxygen and CO2 concentrations, basically during exercise or stress. So oxygen deficiency in cells or high CO2 concentrations in the blood are detected by chemoreceptors, typically in the arteries. This would result in the release of nitric oxide. What nitric oxide does is that it dilates blood vessels. It makes the lumen larger. This would increase cardiac output by allowing an increased stroke volume. Basically, more blood gets pumped to the muscles. So adrenaline is released during scenarios of stress or intense physical stimulation. Like when you're writing a CAPE Unit 2 paper to exam. The hormone stimulates adrenal receptors to stimulate the SAN and the AVN to increase the heart rate. So this is basically like electrical signals. This would thus increase cardiac output because the heart is beating at a faster rate, so more blood would be able to reach the muscles in order to deliver oxygen and glucose for a higher rate of respiration. Blood pressure is the force of the circulating blood on the walls of the arteries. The pulse is a singular force that can be felt against the arterial walls per heartbeat. So the lumens that have a reduced area would result in a higher pressure. So how can it have a reduced area? This would be usually due to a plaque or clot, due to the buildup of lipids and cholesterols. If you have persistently high blood pressure, that is called hypertension. Other factors that affect blood pressure would be exercise or intense exercise. So of course, when you exercise, you are using up more oxygen. So you have low oxygen concentration in your red blood cells. What this does is that it releases nitric oxide and allows the arteries to dilate, and you have increased blood flow and pressure. Cigarette smoking, but the tobacco contains nicotine. Nicotine is a stimulant. It also constricts blood vessels and makes lumen smaller. It would also increase heart rate. Adrenaline, which was explained just before, this is released during stress or excitement. This stimulates the sinoatrial node and the atrioventricular node to increase the heart rate. Lastly, we have atherosclerosis, or the buildup of atherosclerotic plaques. This is the accumulation of cholesterols in arterial lumens, and this would reduce the area of passage for blood flow, thus raising blood pressure. So hemoglobin is a protein in red blood cells with a quaternary structure. That means it has multiple secondary and tertiary structures. There are 4 subunits, and each of the 4 subunits in hemoglobin have heme groups, basically with little iron molecules. Each of these can bind an oxygen molecule. So each hemoglobin can bind 4 oxygen molecules because there are 4 subunits. So when this happens, the hemoglobin is now referred to as oxyhemoglobin. Carbon dioxide could also bind to hemoglobin, but not as readily as oxygen. But when it does, it is called carbaminohemoglobin. So we have to get accustomed with a few terms here. So when we say a gas is dissociated from hemoglobin, it means that the gas is released from hemoglobin, or it unbinds from it, such as when oxygen is released from the hemoglobin into body cells. It is dissociated. Affinity describes how readily the gas binds to the hemoglobin. So since oxygen binds more readily to carbon dioxide, oxygen would have a higher affinity. Carbon monoxide has an even higher affinity than oxygen, so that could potentially fatally bind to hemoglobin, leaving no room for oxygen and thus possibly resulting in asphyxiation. Positive cooperativity. So when an oxygen molecule binds to hemoglobin, the hemoglobin protein experiences a conformational change. That means that it will alter its quaternary protein structure slightly so that more oxygen molecules can bind more easily. Imagine like it folds so it could take in more oxygens more easily. This is to accommodate the more rapid binding of successive oxygen molecules. So it takes less and less time for each oxygen to bind until saturation. So for example, here we have a hemoglobin molecule with no oxygen bound. But as soon as one oxygen molecule binds, remember there's 4 subunits, it takes even less time for the second one to bind, and even less time for the third one, and even less time for the fourth, where it is fully saturated. So remember what the term dissociation means. That means that when the gas is released from the hemoglobin. So if you look at oxygen dissociation, oxygen will only bind to hemoglobin if the oxygen concentration is high enough. Now because air is comprised of multiple gases such as nitrogen, water vapor, oxygen, we refer to the oxygen concentration as a partial pressure. It just means the oxygen concentration. So when there are high partial pressures, so for example in the lungs or the alveoli, more hemoglobin is oxygen saturated. That makes sense because there's more oxygen available, or higher partial pressure, more oxygen is able to bind to the hemoglobin. In low partial pressures, such as around muscle tissues where you wouldn't have much oxygen being stored, you're going to have less hemoglobin being oxygen saturated. In this case here, more oxygen is going to dissociate from the red blood cells, and the hemoglobin is going to go into the muscle cells. So hemoglobin oxygen levels are lower around body tissues due to oxygen dissociation. As I said, it means most of the oxygen from the hemoglobin is being released from that hemoglobin to go into those cells. This is an oxygen dissociation curve. It has a sigmoid shape or an S shape. On the y-axis, we have the oxygen saturation of hemoglobin, the percent, and on the x-axis, we have the partial pressures of oxygen. We'll notice here that in the lungs, we have a very high partial pressure, and this is going to result in a high oxygen saturation in the hemoglobin. This makes sense because at high partial pressures, a lot more oxygen is going to bind to the hemoglobin. Down here, where we have lower partial pressures, we'll see that the oxygen saturation is lower. Because there's less oxygen available, less is going to be able to bind to the hemoglobin. It's as simple as that. So the graph's very steep beginning is due to the rate of oxygen binding rapidly increasing. So the rate here is rapidly increasing due to conformational changes in the hemoglobin. So as one oxygen binds, another one is going to bind as the protein shape changes, and more are going to bind very, very rapidly. So oxygen saturation increases as partial pressure increases. We can see that there, but it's not a directly proportional relationship. So at lower partial pressures, it is more difficult for oxygen to bind. So oxygen dissociation occurs and saturation decreases. So at these lower partial pressures here, what is happening is that because there's less oxygen available, there's going to be less oxygen binding and more oxygen dissociation. So more oxygen is actually going to be released into those cells. So here we have the Bohr effect, and this refers to how hemoglobin's affinity for oxygen is affected in certain conditions. So here we could see the curve shifting to the right. So the curve is going to shift to the right, and basically what this means is that there's a reduced affinity for oxygen. It means that it's not going to take up oxygen as readily. So the curve is going to shift to the right if there's increased carbon dioxide levels, for example near muscle tissue. This is going to decrease oxygen affinity, and this is to allow more oxygen to dissociate from the hemoglobin. So remember, lower oxygen affinity means less oxygen is binding to the hemoglobin, more of it is being dissociated from the hemoglobin to go into cells. So if the curve shifts to the left, for example, this would be when carbon dioxide levels are low, so for example in the lungs. This would increase the affinity for oxygen and allow more oxygen to be taken up and for less of it to be dissociated. Homeostasis refers to the regulation of the body's internal environment in response to stimuli. So for example, if the stimulus is external, so for example like temperature, let's say that I were to go into the desert where the temperature is very high. My body is going to try to maintain my body temperature of 37 degrees Celsius by dilating my blood vessels, by sweating. If I go into a snowfield where the temperature is very low, my body will still try to maintain its normal temperature of 37 degrees Celsius. It's not really going to change, and it's going to help accomplish that by constricting my blood vessels by shivering. So homeostasis tries to maintain equilibrium within the body. It does this by engaging in negative feedback processes. Negative feedback attempts to reverse the change to a set point to achieve equilibrium. So for example, when the body temperature is too high, a person would sweat to lower the body temperature. However, there are cases where you would have loops that would encourage the change. So these are called positive feedback loops, and these would reinforce the change that is happening and allow the process to continue. So example, during childbirth, the hormone oxytocin keeps getting released from the brain. This would allow continuous uterine contractions to push the child out. So many factors are regulated in the body, such as blood glucose levels, salt levels, blood pH, temperature, and each of these has an optimal value or set point. So for example, the body temperature set point is 37.5 degrees Celsius. That shouldn't change, because if the temperature gets too high, enzymes will begin to denature. If it gets too low, there won't be enough kinetic energy in the body. So changes are monitored by receptors or detectors. In the case of temperature, it would be like skin receptors. So receptors transport electrical signals to regulators. In the case of temperature, the regulator is going to be the hypothalamus. This is going to compare this detected value to the set point. So in the body, if the body temperature rises above 37 degrees Celsius, it's going to try to bring it back down. And these are done by effectors. So effectors enact a response to return the value to the set point. Again, as I said, the whole point is to achieve equilibrium. So many of these effectors release hormones to return the factor to the set point. Hormones are transported by the blood and usually act slowly, so there's always a delay between detection and action from the effector. So for example, if my blood sugar level gets too high, insulin would have to be released. The insulin would then work to bring my blood sugar level back down. So if a regulator or control center is damaged, so for example the hypothalamus or the pancreas, there won't be a return to a set point. This could possibly lead to disease and death. Hormones are secretions of endocrine glands that are directly released into the bloodstream and act as chemical messengers. Endocrine glands are ductless, which means that they secrete their substances directly into the blood. They're not like exocrine glands, which secrete their substances through ducts. So for example, sweat glands and mammary glands are exocrine. So hormones act on target cells. These target cells have receptors that are specific to the hormone, and they are typically slow acting, as said before, but they have prolonged or long-term metabolic effects. Here we have a few examples of hormones. ADH or antidiuretic hormone is used to conserve water in the kidneys, basically to reabsorb water back into the bloodstream. Adrenaline is used to increase heart rate in times of stress. Growth hormone stimulates growth in body tissues. Glucagon promotes the breakdown of glycogen to glucose if the blood glucose level is too low. Insulin promotes the conversion of glucose to glycogen if the blood glucose level is too high. Estrogen helps egg cells mature and helps build up uterine lining. Progesterone helps maintain uterine lining on the endometrium. Thyroxine, which is released by the thyroid gland in the neck, regulates metabolism. So the action of insulin. Insulin transports glucose from the bloodstream into the cell cytoplasm, as you can see here. Goes right through the cell membrane. Insulin is secreted by the beta cells in the pancreas's islets of Langerhans as a group of tissues there. Insulin is secreted in response to a rise in blood glucose levels. So for example, if you used to eat a sugary meal, that glucose is going to end up in the bloodstream, is going to get absorbed into the bloodstream, and it has to be assimilated into the cells. What insulin is going to do is going to bind to receptors on the cell membranes that's going to open little gated channels here to allow glucose to enter. Remember, glucose is a very large molecule that cannot diffuse through the bilayer, so it has to undergo facilitated diffusion through these channels here. So glucose can be transported into the cell cytoplasm, and when it ends up inside the cell, there's less glucose in the blood, and thus that would reduce blood glucose level. So if the cell membrane is a door, insulin is a key that would open that door for glucose to enter. In liver cells, the doors, which are the protein carriers, are already on the cell membrane, so it's simple. There's an enzyme called glycogen synthase, and that's going to convert glucose into glycogen. In muscle cells, is a little different. What's going to happen here is that the doors are GLUT4 proteins, and when insulin binds to the receptors, those doors would have to be transported via vesicles to the cell membrane, and then these proteins here would appear on the cell membrane. They would open, and then glucose can enter. So think of it as insulin summoning the GLUT4 transport proteins to allow intake of glucose. Glucagon works the opposite of insulin. So as insulin is used to reduce blood sugar level, glucagon is used to raise it. So this would be like if you're fasting or if you missed a meal or something like that, you are able to actually take the glycogen and any other materials such as amino acids that are stored inside the liver, break those down or convert it into glucose to secrete it back into the blood so it can go into the cells, so you can have sugars for respiration. So glucagon is secreted by alpha cells in the pancreas when blood glucose levels drop. Remember, insulin is secreted by beta cells. It is used to break down glycogen in the liver into glucose to return the blood glucose level to its set point. But it may also convert amino acids and lipids into glucose. This is called gluconeogenesis. Basically, this means making new glucose. So glucagon in the blood binds to a protein receptor in the liver's cell plasma membrane. This would hydrolyze the protein and produce an enzyme called cyclase. Basically, it would break down that protein, a little fragment will pop off, and that would produce this enzyme. Cyclase converts ATP to a secondary messenger called cyclic AMP. So this secondary messenger, cyclic AMP, this is going to break sugar-specific glycosidic bonds in the glycogen to form glucose monomers. Remember, glycogen is a polymer, a very highly branched polymer that is made up of many different glucose subunits. So if you look at this diagram here that shows the negative feedback loop for blood glucose concentration. So here we have the set point, which is the typical blood glucose level. If we have an increase or decrease, the detector is going to pick it up. And this is going to be the alpha and the beta pancreatic cells. There's going to be the regulator, so the alpha and the beta cells are going to compare the value with the set point. As you're going to see if it's too high or if it's too low. If it's too high, it's going to secrete insulin. If it's too low, it's going to secrete glucagon. So how does it work? Insulin would convert glucose into glycogen, and then the blood glucose level would decrease back to the set point. Glucagon is going to convert glycogen into glucose, release it back into the bloodstream, and it's going to cause blood glucose levels to increase. Feel free to pause the video to review the diagram. The diagram is actually quite important. Fruit ripening. So this occurs when a fruit's internal components have fully developed. There's an increase in edibility and ease of consumption. So in the case here, the ripe banana is a lot softer, it's a lot sweeter. The fruit cell walls break down and complex sugars are hydrolyzed. This would increase the sweetness. So this is going to break down all the starch in the unripe banana into sucrose. So the ripe banana is actually softer as well due to pectinase breaking down the cell walls. It is sweeter due to the amylase and sucrase breaking down sugars into sucrose and glucose. These spots here are caused by ethylene. We're going to get into that just now. So also, when ripening occurs, it's going to produce aromatic compounds. This is going to instill scent and flavor. There's also going to be a change in color. Usually what's happening here is that the chloroplasts are being converted into chromoplasts that produce these different colors. This here would make the fruit more attractive to animals and more likely to be consumed. Remember, animals usually eat fruits, digest their seeds, and disperse their seeds through their feces. So ethylene is a gaseous plant hormone responsible for fruit ripening. It can undergo simple diffusion through a large area and influence ripening on adjacent fruits. It remains a gas at low temperatures, which means it has a low melting point. This makes it very suitable to put in air-conditioned warehouses, for example. So thus, a large amount of unripened fruits can be quickly ripened by placing them in a warehouse or container with ethylene-producing ripe fruits. For example, if you look here, if a ripe banana is placed in a bag with another fruit such as a mango or avocado, the ethylene that is being released from the ripe banana will be retained within this plastic bag in a high concentration, and this would accelerate the ripening of the other fruits here. Of course, this makes ripening occur faster, which would increase profits for farmers. Let's look at this graph for ethylene and respiration. On the y-axis here we have the milliliters of carbon dioxide being released per hour, and here we have on the x-axis the days of ripening. So we can see that the rate of respiration increases as ethylene production is increased up until ripening has ceased. So the blue here represents carbon dioxide and the red here represents ethylene. So if you look at the ethylene here, we can see that ethylene is being released mainly after day 2. There's a steep increase here. At day 4, this is when the fruit has completely ripened. The ethylene level plateaus. But if you look at days 2 and 3, this is where the levels of ethylene are going up, but the level of carbon dioxide has a very steep increase. This is because the rate of respiration is going up as the fruit is ripening. A few days after ripening, however, cell death and spoilage will occur. Excretion is the removal of bodily waste from products of metabolism. So examples would be urea, carbon dioxide, and water. Accumulation of these waste products here in tissues will become toxic and will of course poison the person who has them accumulated. Urea is the main waste product formed from deamination, which is the removal of amino groups from amino acids. If you look at the diagram on top here, we can see that this is an amino acid. We have the carbon molecule connected to a residue, a hydrogen atom, a carboxyl group, and the amino group here. During deamination, this group is removed and this forms ammonia. Ammonia is highly toxic to cells. So what is happening here is that this ammonia, through this cycle called the ornithine cycle, which you don't have to memorize all these different parts, but what you have to know is that the ammonia, which is toxic to cells, is converted to urea, which is much less toxic to cells, and this is excreted in the urine. So the ornithine cycle in the liver helps convert toxic ammonia to the more soluble so that it can be dissolved in urine, and much less toxic urea. So here we have the external gross kidney structure. Here we have the renal artery, which is going to carry blood from the heart to the kidneys. The renal vein that's going to carry blood away from the kidneys and back to the heart. We have the renal cortex, and this is the outer part of the kidney. This is going to contain Bowman's capsules and convoluted tubules of the nephrons. We have the inner part of the kidney, which is called the medulla, and this contains collecting ducts and loops of Henle. The ureter is going to transport any urine to the bladder, and the renal pelvis is going to collect the urine flowing into the ureter. Remember, the whole point of the kidney is to excrete waste and to form urine to be excreted from the body. Here we have the kidney nephron. So the nephron is the functional unit of the kidney. They help with ultrafiltration, selective reabsorption, and osmoregulation. Ultrafiltration is the buildup of capillary pressure, which is going to force small molecules into the glomerular capsules to form a filtrate. That's why you're seeing these molecules flowing through here. Selective reabsorption is going to remove any nutrients or any useful substances such as glucose from the filtrate to be put back into the bloodstream. This is why you're seeing that some of it is moving into the capillaries here. This would be like amino acids, some sodium, some glucose, and so on. Lastly, we have osmoregulation, which is the conservation of water in the blood due to the hormone ADH, which is antidiuretic hormone. Basically, the whole point of the kidney nephron here is to get small soluble substances to form a filtrate coming in through the Bowman's capsule and the tubules here. At the end here, we are supposed to form urine, which is basically urea, water, and any mineral salts such as sodium. Let's take a closer look at the kidney nephron. First, we have the afferent arteriole. Think of A as arriving, and this is basically bringing blood into the glomerulus, arriving. Then we have the efferent arteriole. This is going to carry blood away from the glomerulus. So think afferent for arriving and efferent for exiting. Afferent is bringing blood in, efferent is taking blood out. Then we have the glomerulus. The glomerulus is a capillary cluster, and this is going to filter blood to Bowman's capsule. In case you're wondering, the term glomerulus refers to like a ball of yarn, so it kind of looks like that. This whole structure here is called the Bowman's capsule. This is a cup-like sac that accepts the filtrate from the glomerulus. Think of it as a funnel cup. Coming through here, we have the proximal convoluted tubule. This is the site of selective reabsorption. And then coming down here, we have the loop of Henle. We have the descending limb and we have the ascending limb. This is going to regulate fluid and ion composition in the filtrate. Coming down to the end here, we have the distal convoluted tubule and the collecting duct. The collecting duct is going to respond to antidiuretic hormone or ADH, and this is going to facilitate water reabsorption into the blood. So for example, on a very hot day, you want to get water back into the bloodstream so that you can form sweat to cool the body down. In this case here, water is going to move from the collecting duct back into these capillaries that you're seeing here. These are called peritubular capillaries. Basically, it means capillaries that wrap around these tubes. So let's take a look at ultrafiltration. Here we have the afferent and the efferent arteriole. Remember, afferent for arriving, efferent for exiting. The efferent arteriole lumen is smaller than the afferent. If you remember anything about the lumen being small, is that is going to build up pressure. So the hydrostatic pressure is going to build up inside the glomerulus here. This pressure is going to cause water and small solutes such as urea, glucose, and amino acids to flow from the glomerulus into the capillary endothelium and through the Bowman capsule basement membrane. Think of that as a filter that's going to push everything through the nephron here. Anything that is too big to end up as a filtrate is just going to exit through the peritubular capillaries. So for example, you're not going to get any red blood cells or white blood cells being pushed into here unless the kidney itself is very damaged. Next, we have reabsorption. So reabsorption typically occurs in the proximal convoluted tubule or the PCT. So what's happening here is that the proximal tubule epithelial cells have numerous villi, as we're seeing here, and very thin membranes and also many mitochondria to facilitate active transport. What's going to happen here is that they're going to export ions and water back into the capillaries, that's what you're seeing here, via diffusion and active transport. Of course, it's going to expend ATP to do this. So when sodium ions and potassium ions are actively transported, glucose is co-transported with it. Think of sodium opening the gate, and amino acids and glucose can also go through that same gate. So glucose is co-transported in a symport manner along with sodium ions as they are transported against a concentration gradient into the capillaries. So to sum it up nicely here, sodium and potassium open the doors, amino acids and glucose can then rush through and end up back into the bloodstream, selectively reabsorbed. The loop of Henle helps with continued water reabsorption into the blood. It has a descending limb, which is permeable to water, and an ascending limb, which is impermeable to water. Sodium and chloride ions are actively transported from the ascending limb, that's what you can see here, to cells that are surrounding the descending limb. So as these ions are being transported out of the ascending limb, what's going to happen is that is going to lower the water potential around this area. There's going to be a higher water potential inside the descending limb and a lower one outside. This is going to allow water to flow into the surrounding cells here, of course with those ions. So the water is going to move into the surrounding cells, or is going to move into the capillaries around those cells. So this is called a countercurrent type of flow. The rest of the filtrate, by the way, would continue to the collecting duct. So at the collecting duct, the filtrate here is basically urine. This is unabsorbed water, urea, and any dissolved mineral salts. ADH is a hormone secreted by the posterior pituitary gland during time spent in hot environments or dehydration. So for example, if you're out in the hot sun, your body is going to secrete ADH and it's going to tell the kidney nephrons to absorb more water back into the bloodstream. ADH facilitates the reabsorption of water from the collecting ducts into the peritubular capillaries in the bloodstream. This is what you're seeing here. ADH interacts with the collecting duct plasma membrane. So what this does is that it's going to transport aquaporins from the cytoplasm to the membranes. Aquaporins, remember, are water-filled protein channels. These allow water to flow in and out. So these aquaporins actually increase the permeability of the duct. This would allow water to be reabsorbed to cool and hydrate the body's cells. In cooler environments, ADH is suppressed and the collecting duct remains mostly impermeable because there's no aquaporins being brought here. So in low water potentials or hot temperatures, ADH secretion is stimulated, the collecting duct walls increase the permeability, aquaporins bind to the surface membranes, water flows out of the collecting duct, and they get a small volume of concentrated urine. In high water potentials or in low temperatures, ADH secretion is suppressed. The collecting duct walls remain impermeable to the water because there's no aquaporins on the surface membrane. So all the water basically stays within the collecting duct. And there's a larger volume of dilute urine. Basically, the urine is going to be more clear colored. Here we have a negative feedback loop of the same thing. Feel free to pause the video and take a look at it. Now let's look at the kidney tissue histology. First are the transverse sections of a kidney medulla and the kidney cortex. If you have to look for glomeruli, they're very simple to locate. They're large and globular. They look kind of brain-like in my opinion. Now in the medulla, you're going to see the collecting duct. The cell borders in the collecting duct are clearly visible and they bulge towards the lumen. This is basically where the aquaporins are going to be located. The loop of Henle cell borders are not clearly visible. Here you can see them, one here and one here. We have the thin segment and the thick segment. The distal convoluted tubules appear to be slightly denser or darker than the proximal ones. Here you can see a proximal convoluted tubule here, and here you can see a distal convoluted tubule here. Now what if you end up with proteins and glucose in the urine? Well, first of all, proteins are too large to pass through the Bowman's capsule basement membrane. But if you have enough blood pressure, very high blood pressure, you have membrane damage in the nephrons, they can be present in the urine. The implied diseases here are kidney failure and severe hypertension. If you end up with glucose in the urine, you're actually supposed to have none. This is going to imply that capillary blood glucose concentration is exceedingly high, and this is going to limit the transport of blood glucose from the proximal convoluted tubule. So basically, not enough glucose is going to make it back into the bloodstream. In fact, all of it is supposed to make it back. So if you end up with glucose in the urine, the implied disease here is going to be diabetes mellitus, whether it's type 1 or type 2. The nervous system consists of the central nervous system or CNS and the peripheral nervous system or PNS. The central nervous system comprises the brain and the spinal cord. The peripheral nervous system comprises the peripheral nerves. That's basically all the other nerves around the body. Electrical impulses or action potentials are very rapid messengers of sensory input from receptor cells and carrying instructions to effectors such as muscles or glands. Basically, they are faster acting than hormones. Neurons are cells which make up these pathways. So there are three types of neurons: sensory, motor, and relay or interneurons. Relay neurons connect sensory to motor neurons. On the diagram here we have a motor neuron. At the end we have the cell body or the soma. These contain organelles such as the nucleus or mitochondria. Branching out from these are the dendrons and very small dendrites. These branch out from the cell body and they propagate impulses received from other neurons. You could think of them as connection points. The axons are long processes that come out of the cell body of the neuron along which impulses are conducted. Think of this as like a wire that would conduct electricity. We have Schwann cells that wrap around the axon to produce a myelin sheath. Myelin is a protein that acts as an insulator, and gaps between these cells are called the nodes of Ranvier. We'll get more into those just now. At the end of the motor neuron, they are the axon terminals. These would be the connection points to other neurons here. Here we have a sensory neuron. So sensory neurons are found on the sense organs, such as the skin or the retina. They detect stimuli and pass them on to the central nervous system to be interpreted. Motor neurons are connected to effectors to facilitate movement or responses to stimuli. Remember, motor comes like movement, it's mostly connected to muscles or glands. Sensory neurons are similar in structure to motor neurons, but the cell body is located in the middle of the axon instead of at the end. Resting potential. Now imagine that you're not doing anything. you would expect there to be no charge in neurons, but in fact neurons are still active even at rest. It tries to maintain a membrane voltage of minus 70 millivolts. If you used to look on the diagram here, imagine that this here is an axon, right? A section of the axon. So inside the axon here, we would have a negative voltage, negative 70 millivolts. So it's slightly negative on the inside compared to the outside. So inside the axon is negative while outside the axon is positive. So the axon is said to be polarized. So remember, once again, the exterior is positive because there are much more sodium ions there. A smaller amount of potassium ions inside. An easy way to remember it is that, remember, bananas contain a lot of potassium and, well, salt contains sodium. So think of it as a salt-covered banana, where inside there's more potassium ions and outside there's more sodium ions. Now, once again, remember, the interior is negative and the exterior is positive. So basically, why outside has more positive charges is because of these things called sodium-potassium pumps. What happens here is that these allow 3 sodium ions to leave for every 2 potassium ions that enter. So there's a net change in charge here because every time 3 sodium ions leave, 2 of these potassium ions enter. Of course, this uses ATP. Now we also have channels, leaky potassium channels on the cell membrane that allow potassium ions to exit. So even more positive charges end up outside. So we'll have channels where potassium ions would just exit. So the electrochemical gradient maintains this resting potential of negative 70 millivolts. So remember, more of the positive charges are leaving here. So outside here is positive and the interior is negative because there's less positive charges inside here. So what is an action potential? So an action potential is an event that coincides with the transmission of an electrical impulse. They occur when nerves are not at rest, basically when the nerves are experiencing action. So an action potential occurs in 3 main phases: depolarization, repolarization and hyperpolarization, and impulse transmission. Let's take a look at depolarization. Well, you better hang on to your seats because this could get a little complicated. Okay, so when depolarized, the axon interior will have a voltage of 0 millivolts. Now remember, inside here, this is the interior of the axon. Inside here at rest is negative 70 millivolts, slightly negative, and outside here is positive. If it was to get depolarized, you would expect here to have no charges. So how can we have no charges? How can we turn a negative charge into no charge? Well, we need to bring in positive charges. So the axon becomes depolarized when these positive charges here surge into the axon. So these positive charges here are sodium ions. So how does that happen? Now imagine that, this axon is in, near your skin and somebody was to touch your skin. Now that would be applying pressure to the skin. So a lot of these channels here are voltage gated or mechanically gated, which means that they open due to a stimulus. So channels permeable to sodium ions open due to stimuli such as pressure. So the channels will open and these sodium ions would rapidly diffuse through and enter the axon cytoplasm. This here would neutralize these negative charges and inside here would become neutral. It would become 0 millivolts and that would be as depolarized because previously it was polarized. Now there's a problem here now. What happens is that when this gate opens, too many sodium ions end up rushing in. Of course, so now it's not going to be 0, it's going to be slightly positive. So the voltage is now about 30 millivolts, which is more than 0 millivolts. So that there is called overshooting. Too many sodium ions end up rushing in and instead of it being neutral, it becomes slightly positively charged. So with repolarization now, we are trying to get both sides of the axon polarized once again. Where inside here would be negative and outside here would be positive. Remember, currently now, inside here is positive because all these sodium ions would have rushed in. Outside here would have been negative because it would have lost a lot of these positive charges because they would have rushed into the axon. So there are now more positive charges inside the axon, but the axon needs to return to resting potential, which is negative 70 millivolts. So we need to get these positive charges out of the axon now. So it's going to flip back from positive back to negative. So how does that happen? Well, they have voltage gated potassium channels. These are going to open due to the change in voltage inside here. And this is going to allow positive potassium ions to move out. So these potassium ions are going to move out here. And this is going to lose the positive charges, and it should go back to negative, which is resting potential. So the axon interior will once again become negative. But something happens here. It becomes a bit more negative than resting potential, about negative 80 instead of negative 70 millivolts. This is called hyperpolarization. Basically what happens here? Well, a little more overshooting happens here because a little more positive ions end up exiting than they're supposed to. But I shouldn't say supposed to, because really this is very helpful, because what happens is that this gives it a little time to return to resting potential. It doesn't bring it back to resting potential immediately. It gives it a little time, and that period of time is called the refractory period. Because what's going to happen now is that sodium potassium pumps are going to try to work to get this negative 80 millivolts back down to negative 70. That is fine, because an action potential cannot take place during this refractory period. This is helpful because if action potentials occur too often, it could end up damaging the neurons, or it could cause a lot of pain for the person who has this condition. So there must be some time between sending the signals of these action potentials. It can't occur just all at once. Alright, so here's outside the cell and here's inside the cell. And a way to think of depolarization as a store opening for Black Friday. And you have this big Black Friday crowd waiting outside, and as soon as the door opens, too many people are gonna rush in at once. So this is like overshooting. So where you're gonna have all these sodium ions rushing in through this channel here when the doors open. So this is like overshooting, where there are much more positive ions inside the axon than expected. And instead of really depolarizing it, it's gonna just polarize it in a different way. So instead of here being 0, it's gonna turn from negative 70 millivolts to positive 30 millivolts. And think of repolarization now as the guard saying, hey, too much oil in the store, we need to open this door and send some oil out. So there's the guard chasing some of the crowd out of the store. So that's what you're seeing here. These potassium channels are going to open due to the changes in voltage here, and it's going to let out some of the potassium ions. The sodium potassium pumps are going to help get rid of any extra positive charges here, because inside the cell has to now become negative once again. So impulse transmission is when the electrical impulse is transmitted along the axon like an energy carrying wave along the surface of the ocean. The wave itself consists of a series of depolarizations. So one depolarized region impacts the successive region, and that becomes depolarized. So this would continue until the impulse reaches its destination, such as an effector like a muscle or a gland. So here we can see inside the axon would be normally negative at rest. But this would be the series of depolarizations, where these charges here are going to continuously flip. Alright, so this graph looks pretty complicated, but we're going to break it down. So this is graph in action potentials. Now you have to remember here is not all changes produce an action potential. So if the stimulus is way too small, for example, a very, very, very light touch, it may not activate enough sodium channels to cause depolarizations. Thus no action potential develops. This is called a failed initiation. Basically, the body is saying that this change is too small, we don't need to respond to this change. So it said that the threshold for an action potential is about negative 55 millivolts. Only around then you will get enough voltage gated sodium channels to be activated. So let's look at the graph now. Here we have the stimulus, and let's say that the stimulus is like somebody touching your skin, somebody touching your hand, for example. What's going to happen? Now remember, all this is color coded here. So the sodium channels are going to open as a response to that pressure, that touch, and depolarization is going to occur. Now remember, depolarization is going to seek for the voltage to be 0. So sodium ions are going to rush into the cell when this channel opens. It's supposed to be 0, but instead it ends up overshooting into the positive region, so about a positive 30 millivolts. Around here is where the sodium channels are going to close. and potassium channels are going to open due to changes in this voltage here, and repolarization is going to occur to get some of those positive charges out. So that's why the voltage here is going to drop. So the potassium channels are going to open, charges are going to flow out, so it's going to go back to rest, but it's going to go a little past negative 70, where the potassium channels are going to close a little past negative 70 until about negative 80. Basically, what's going to happen at the refractory period here is that sodium potassium pumps, remember what they do is that they take in 2 positive potassium ions for every 3 positive sodium ions that they let out. So this is going to take a small while to build it back up to negative 70 millivolts. So this little dip here, this is called hyperpolarization. So the frequency of action potential. So a weak stimulus, for example a soft touch, generates a low frequency of action potentials in just a few neurons that are stimulated. But a strong stimulus, for example a slap, generates a higher frequency of action potentials in many neurons. Saltatory conduction. So first let me explain what the term saltatory means. Saltatory refers to leaping, like how a frog would leap. So in a myelinated axon, the action potentials are conducted in a high velocity transmission, as the spread of depolarization occurs in leaps across the nodes of Ranvier. So you see here, this is the Schwann cells. The nodes of Ranvier are these little gaps here. So what's going to happen is that imagine like it's going to leap like this. So this is saltatory conduction. So in an unmyelinated axon, the velocity is much lower. It cannot get to leap from gap to gap, as the action potential must be propagated from cell to cell along the axon in a continuous progression instead. So synapses are sites that lie between 2 successive neurons. So here we have the presynaptic bulb or the presynaptic neuron, and this would be the postsynaptic neuron. And the little gap here, this is the synaptic cleft, and this would separate both these neurons here. This gap is very, very, very small, but what would help to get the action potential to be conducted across would be a chemical neurotransmitter called acetylcholine, and this would allow the propagation of the action potential across the synapses. Think of the acetylcholine as a bridge between these two. And by the way, a synapse located between a motor neuron and a muscle fiber is called a neuromuscular junction. So how do we get these action potentials across these synapses? How do we help it propagate? How do we let it cross this bridge? Now this is the presynaptic bulb, and down here is the postsynaptic membrane. Between here is the synaptic cleft. First of all, the action potential is going to arrive from the axon to the presynaptic bulb. What is this going to do? This is going to trigger the opening of sodium and calcium channels. And so it's going to allow sodium ions to enter and calcium ions to enter the bulb cytoplasm. What's going to happen in response to this is that the acetylcholine here, this is going to get packaged into vesicles. So when they get packaged into vesicles, these vesicles are going to drift towards the synaptic cleft. And via exocytosis, they're going to fuse with the plasma membrane here, and they're going to release the neurotransmitter acetylcholine. That's what you're seeing happening here too. So on the postsynaptic membrane now, we're going to have all these acetylcholine molecules here. And on the postsynaptic membrane, the acetylcholine is going to bind to receptors on little protein channels here. And this is going to allow the influx of sodium ions. This is going to depolarize the postsynaptic membrane. And this depolarization is going to allow the action potential to propagate from the presynaptic bulb to the postsynaptic membrane. That's what's going to bridge this here, this change in charge that's going to happen in the postsynaptic membrane and in the presynaptic bulb. This is what's going to transmit the impulse. So what happens after that though is that we're going to have an enzyme that is called acetylcholine esterase, and that's going to break down acetylcholine into acetate and choline. That's what you're seeing happening at 4. The choline is going to return to the presynaptic bulb and is going to reform acetylcholine. So this is how this will be recycled. So this is kind of like a cycle. So the choline is going to join with acetyl coenzyme A to become acetylcholine. Health and disease. Well, health is defined as a complete state of physical, mental, and social well-being. This is according to the definition by the WHO, the World Health Organization, whereas disease is the impairment of the function of the body and the mind. So diseases can be communicable or non-communicable. Communicable diseases can be spread from one individual to another. They are infectious and they are caused by pathogens such as bacteria, viruses, fungi, and protozoa. Non-communicable, on the other hand, cannot be spread from one individual to another. They may be caused by a lifestyle or be inherited. Example would be diabetes, which cannot be transmitted from one person to another. It can transmit the risk from a parent or offspring, but it can't actually transmit the disease itself. So there are many other categories of disease. However, it is important to keep in mind that any one disease cannot be placed in a single category. So example would be diabetes. Diabetes is classed as a physiological disease because it is caused by a malfunction in an organ or system, but it's also classed as a chronic disease, which is a disease that is long-term. We have other diseases as well. So if you look at diabetes, this is caused by malfunction in the pancreas. And other examples would be coronary heart disease and asthma. Mental diseases occur due to the impairment of the mind or the tissues within. Examples would be Alzheimer's disease, clinical depression, and schizophrenia. Other chronic diseases would be chronic bronchitis and, of course, asthma. Infectious diseases are those caused by the introduction of pathogens to the body. So a virus causes dengue fever, a protozoan causes malaria, and bacteria causes cholera. Social diseases are caused by an individual living or working within an environment prone to contamination by pathogens or having no access to a balanced diet. For example, both cholera and dysentery are caused by bacteria that are usually found in water. Cholera is usually transmitted by flies. So there's a lot of flies in the area, you're going to have a higher incidence of cholera. Scurvy, rickets, and night blindness are due to deficiencies of vitamins or minerals. So scurvy is due to a lack of vitamin C, rickets due to a lack of vitamin D or calcium, and night blindness is a lack of vitamin A. Degenerative diseases are caused by the gradual loss of function of its tissues and organs. Alzheimer's, which is of course due to the degradation of brain tissue, and osteoarthritis, which is due to the degradation of cartilage. Deficiency diseases occur due to a lack of a particular nutrient in the diet. So we have kwashiorkor, which is due to the lack of proteins, scurvy and night blindness due to the lack of vitamins C and A. Hereditary diseases are caused by inheriting alleles, typically recessive alleles, that would translate to malfunction in proteins. Examples would be sickle cell anemia, hemophilia, and cystic fibrosis. All of these diseases you would have looked at in Unit 1. Self-inflicted diseases, also referred to as lifestyle diseases, because typically they occur due to a bad habit. So these occur due to the intake of excess nutrition or drugs. So liver cirrhosis or liver scarring, it could be due to overconsumption of alcohol. Lung cancer could be due to a bad smoking habit or working in an environment with a lot of smoke. And coronary heart disease due to the overconsumption of lipids and cholesterols. You have to also learn how to analyze disease data. So if you look at the y-axis on this graph here, this shows the number per 100,000 of population, and this here shows the year from 1982 to 2010. We have three different line graphs here, which is HIV cases, AIDS cases, and AIDS deaths. So what are some observations that we can make as time passed? Well, first of all, we can say that the number of HIV cases generally increased from 1982 at least to 2003, which would be around here. And there'll be small dips and fluctuations along that time period. One such dip would have been around 2001, which would have been around here. And after 2007, which will be here, there was a sharp decline in cases. This is most likely due to increased awareness. You have a lot more people practicing safe sex. You have antiviral technology, so this would have reduced HIV cases. Other observations that we can see here. So in terms of the AIDS cases, you can notice that this was always less than the HIV cases. This is due to the dormancy period before AIDS developed. Not everybody who have HIV is going to develop AIDS. Or some cases with HIV may die before AIDS develops. Lastly, if we look at the AIDS deaths, we can see that the AIDS deaths are lower than the AIDS cases. This could be especially after 2001, and this could be possibly due to earlier detection or any opportunistic infections that may pop up. You could get antibiotic treatment for it. An immune response is defined as the reaction of white blood cells to the presence of a foreign substance not recognized as a constituent of the body. So if something foreign comes into the body, such as a toxin or a pathogen, the immune system is going to recognize it and try to eradicate it. So these foreign substances contain structures that are called antigens. So say here we have a bacteria, the bacteria is going to have antigens on them. So think of the antigen as something that's going to trigger the body's burglar alarm. It's going to sound the signal and allow the white blood cells to come and enact an immune response. So we have phagocytes and we have lymphocytes. So phagocytes patrol the bloodstream in search of antigens. When they encounter them, they engulf them. Think of them like they eat them. They are usually short-lived. So when they engulf these pathogens, they can then present them to other white blood cells. You'd be like, hey, this is what it was. Go and find ones that were similar to this. So they can present these antigens that were engulfed to lymphocytes. We have two types of lymphocytes. We have B lymphocytes and we have T lymphocytes. B lymphocytes or B cells multiply and secrete antibodies. These are going to counter antigens. T lymphocytes can be T helper cells, and these could stimulate B cells and phagocytes, or they can be T killer cells or cytotoxic cells, and these will just destroy the cells infected by the pathogen. So due to all these different types of white blood cells, we have two different types of responses. We have the humoral response and the cell-mediated response. Humoral responses involve B lymphocytes. This is where you can see here, where we have a phagocyte that is engulfing a virus and thus the antigens, and now it is called an APC or antigen presenting cell. It can present this antigen to a T helper cell, which can then send signals to either T killer cells, or the B cells are going to produce antibodies, and this is the humoral response, and the T killer cells are just going to kill the infected cells. This is called the cell mediated response. Here we can see the phagocyte acting as an antigen presenting cell to a T helper cell, which is now going to recognize the antigen and send the signals to the other white blood cells to take action. So let's compare B lymphocytes and T lymphocytes. So B lymphocytes originate in the bone marrow, so do T lymphocytes. But B lymphocytes mature in the bone marrow and T lymphocytes mature in the thymus gland. And this is why it's called B lymphocytes for maturing in the bone marrow and T for thymus gland. The B lymphocytes enact the humoral response, which is a more rapid response. The T lymphocytes enact the cell-mediated response, which is a more slower, delayed response. B lymphocytes secrete antibodies. T lymphocytes secrete cytokines from T helper cells. B lymphocytes differentiate into antibody-secreting plasma cells and memory cells that would retain the data from the pathogens. T lymphocytes would differentiate into T killer cells, T helper cells, and memory cells as well. So let's take a closer look into the humoral or the B cell response. First, the antigen receptor in the B cell is going to bind to the specific complementary antigen. Remember, the antigen would have been from the virus or the bacteria, whether it's like a spike protein or a little piece of the bacteria that the B cell could recognize to take action. What's going to happen next is that clonal proliferation is going to occur. These B cells are going to undergo cell division, and they're going to divide to produce a large number of B cells. This is called clonal proliferation. Next, what's going to happen is that because you have a lot of these plasma cells and a lot of these B cells are differentiated into plasma cells, you're going to get a large amount of antibodies being produced to fight off what would have brought in these antigens, bacteria, the viruses. You're also going to get memory cells that are going to produce antibodies if a future infection occurs. So the B cells experience a specific binding response, which means that the antigen must be in contact with the receptor on the B cell surface. So think of it as similar to the enzyme's lock and key mechanism. The antigen binds to these receptors to allow proliferation to happen and then differentiation into either plasma cells or memory cells. Now let's look at the cell-mediated or the T cell response. First, the phagocyte is going to act as an antigen-presenting cell. What it's doing is showing the antigen to the T helper cells. That's what you're seeing here. It engulfed the virus, and now it's showing in A, this is what the viruses look like. So T cells are going to experience clonal proliferation as well. So they're going to divide, and differentiate to form many T helper cells and T killer cells. So T killer cells are going to attempt to destroy the infected cell with the virus in it. Remember, these are also called cytotoxic cells. The cell will eventually undergo lysis, so the cell is going to get totally destroyed, and then memory cells are going to be produced, and these will proliferate upon future infection. So if one of these future antigens was to be spotted, so if one of these bacteria or viruses was to come to the body in the future, was going to have these memory cells are going to remember, and these now are going to present these antigens to the cell, so it's going to have a much faster response. So let's recap here. Pathogens are like enemy soldiers. T killer cells are the melee combatants. They get very close. B cells are like snipers, and the antibodies are like the bullets. T helper cells are like messengers. Antigen presenting cells are like spies or recon. They get information and they show it to other soldiers. Memory cells are like veterans who have already fought this war and they have already seen the enemy, but now they can form strategies on how to very quickly eradicate the enemy. So what happens during phagocytosis? So phagocytes patrol the bloodstream and surrounding tissues using plasma membrane extension or pseudopodium. Pseudopodium is Latin for false foot. Basically what it does is that it extends pieces of its plasma membrane in order to move. Think of it like it's crawling through the bloodstream. So what they do is that they ingest pathogens via endocytosis by evaginating or wrapping their plasma membranes around them to form a phagosome. That's what you're seeing happening here. They would form a little fold or evagination, and this is going to wrap around the pathogen. You see here is going to form a little vesicle, and this is called a phagosome. Next, what's going to happen is that lysosomes are going to secrete enzymes to digest the material. So lysosomes are going to digest the bacteria, is going to break it down the organic material. The phagosome is going to fuse with the lysosome, and then the lysosomal enzymes digest the captured material. Finally, the digested products, some of them might be absorbed, but a lot of them are just going to be released via exocytosis. So the same vesicle is going to fuse with the plasma membrane here and release all this material back into the bloodstream so that, the liver could get rid of it. So during all of this, you're going to have an inflammatory response. So imagine like you get stung by a mosquito and you see the area swell up a little bit. This is due to cytokines, and cytokines are chemicals that sometimes stimulate the immune system. What these do is that they interact with blood proteins called complements. This would enable a cascade of reactions which are going to activate enzymes that would lead to inflammation. You're also going to have mast cells. So mast cells release histamine. Histamine would dilate capillaries and ease blood flow to the inflamed location. Imagine like it opening up the road to allow white blood cells to drive through or flow through. This aids in the transport of white blood cells to infected tissues. So this is the use of inflammation. As a result, this is where the person might get a fever, or you might get a lot of heat being released from that particular area. Antibodies are blood glycoproteins secreted by B lymphocytes during the humoral response. We would have gotten through that already. So here we have the structure of an antibody, and the antibody actually has two regions. The variable region, this will differ from antibody to antibody, and the constant region, which will be the same for every antibody. So each antibody has a specific region to attach its complementary antigen. This is the variable region. So this has an antigen binding receptor. Again, think of this like an enzyme with a specific active site, like a lock and key mechanism. This part here would be different for each different antigen. So you'd have one for the dengue virus antigen, you'd have one for the cholera antigen, you'd have one for the tuberculosis antigen, and so on. So and then we have this constant region. So this constant region, which is common to all antibodies, this consists of light and heavy polypeptide chains connected by disulfide bonds. If you were to get infected by a pathogen, you would first have a primary response. If you were to get reinfected later on, you would then have a secondary immune response. So what happens during the primary immune response? Well, this occurs due to initial exposure to an antigen. This is like the first time, for example, you was to come into contact with measles or COVID-19. This will yield a moderate concentration of short-term plasma cells. So you see in the antibody concentration here, this is going to rise not too much, but a little bit, because this is going to be in response to the infection. So this will produce plasma cells that will then produce antibodies. So the antibody concentration gets higher until the infection has gone away. So remember, around this time here, you're going to get clonal proliferation of a lot of B cells. Some of them are going to become plasma cells, and some of them are going to become long-term memory cells. That's going to store the data. Remember, those already have seen the enemy. So this may occur after a first vaccine dose, or due to just natural exposure to a pathogen, the first exposure. So what happens during the secondary immune response? Let's say that you were to encounter the same antigen or pathogen a second time. We're going to enact a secondary immune response. Remember, you would have built up memory B cells from these differentiated B cells that would occur due to the initial response. So these memory B cells now, they would have already remembered what the antigen, looked like. And the B memory cells can be differentiated into a greater number of plasma cells. So you're going to get a lot of these proliferating to form more plasma cells because they're already there. They already know what it looks like. We could get rid of it faster. Of course, more plasma cells means that more antibodies can be produced. This is why the antibody concentration goes way up. So there's much more rapid proliferation of antibodies and a much shorter time between exposure or detection of the antigen. So it gets rid of the pathogen in a much shorter period of time. This could be after a second dose of a vaccine or due to a future exposure of the antigen. Monoclonal antibodies. These are antibodies made by cloning a unique B lymphocyte. Now there's a difficulty with producing cloned leukocytes. First of all, cloned B plasma cells do not secrete antibodies. And antibody secreting B plasma cells, they don't divide. So how do we solve this problem? Well, what they do is that they could fuse a B plasma cell that secretes antibodies with a cancer cell. We know that cancer cells are known for dividing. This would form something called a hybridoma. So hybridomas can naturally proliferate and produce a large amount of monoclonal antibodies. To stimulate the production of antibodies, mice were injected with antigens. So we inject the mouse with antigens, and the mouse now is going to produce a lot of cloned antibodies, and this is called monoclonal antibodies. Pregnancy tests also uses monoclonal antibodies. So pregnancy tests are dipsticks that take up urine. Pregnant women would produce a hormone called human chorionic gonadotropin, or short form HCG. If a woman is not pregnant, she's not going to produce HCG because this is usually produced by the blastocyst, which is the future form of a fertilized egg. What happens here is that the woman will urinate on the stick. The urine is taken up by capillary action, and there's 3 different zones here. You can see it labeled 1, 2, and 3. So 1 is the reaction zone. The reaction zone contains monoclonal mouse antibodies, but these here can specifically bind to the hormone HCG. Remember, this would only be in pregnant women, and these have an enzyme that is going to activate a dye. So part 2 now, this is the result window. This is what actually contains the dye. So if there's HCG present in the urine, antibodies are going to bind to the dye. So if there's no HCG, it's not going to bind to the dye. So if there's HCG, it's going to bind to the dye, and this is going to produce a colored line to indicate pregnancy. So 3 is the control window, and this contains anti-mouse antibodies and dye. Now remember, in the reaction zone here, we had mouse antibodies. So part 3 now, we don't need to have HCG reacting with these antibodies here. Once they are mouse antibodies, they are going to react with the anti-mouse antibodies. So it doesn't matter whether or not the woman is pregnant, the reaction is still going to occur here with the dye. So you're still going to get a line here whether or not the woman is pregnant or not. So this is why it is called the control. This is just to show that the pregnancy test is working. So if 2 colored lines form, as you see in the picture here, a woman is pregnant. This is because HCG molecules in the urine bind to the monoclonal antibodies, and this will activate the dye. This will basically be this dye here. If 1 colored line forms, a woman is not pregnant. Anti-mouse antibodies, which are not specific to HCG, activate the dye here. This is the control. And if you get no lines forming, that means the pregnancy test is faulty. It means that the enzyme did not activate the dye at all. It means that you probably have to buy a next pregnancy test. Let's take a look at the diagram above. Which reaction would occur at the reaction zone, the result window, and the control window? We have reaction A, reaction B, and reaction C. And we have a little key here to show which molecule is which. Feel free to pause the video to figure it out. So these are the answers here. A would be in the control window, B would be in the reaction zone, and C would be in the result window. Feel free to pause the video to read the explanations. So what are the different types of immunity? Well, immune responses happen due to exposure of foreign substances or antigens that are attached to pathogens or toxins. This immunity occurs due to the production of antibodies from B cells. So this can be passive immunity, which is seen here. This is where if antibodies are introduced from an external source. So for example, monoclonal antibodies if you was to inject them, or breast milk. This would be if the body did not form the antibodies itself. So active immunity is if the body is allowed to produce its own antibodies and memory cells, such as with a multi-dose vaccine, which is going to stimulate the production of antibodies, or just a natural infection, like if you was to just get measles and you form the antibodies against it. It came from within your own body. You use energy actively to produce antibodies, as opposed to getting it from outside, which would be from passive immunity. Now let's break down the 4 types of immunity. We have naturally acquired active, and this is obtained from the primary response to pathogen exposure. So this is like when B cells develop antibodies and memory cells. This is like if you get COVID-19 just by walking around and you get it from somebody, and then you develop antibodies and recover from it. Natural passive immunity obtained from mother to offspring transfer of antibodies from either the breast milk or through the mother's placenta. So this is like when the baby is born, the baby receives through the placenta some antibodies from the mother, and the baby also receives antibodies from the breast milk or the colostrum. Artificially acquired immunity is the long-term immunization obtained from the primary response due to intentional exposure to the antigen, such as a vaccine. So again, this is like before you get infected, you will get injection of the antigen, or at least a part of the antigen or a part of the pathogen, into your body, and that they will stimulate B cell production, so that if you was to get infected in the future, it would limit how much the infection would actually affect you. Then we have artificial passive immunity, which is a short-term immunization obtained from the injection of antibodies such as monoclonal antibodies. No memory cells are developed. So for example, let's say that you do happen to contract COVID-19, they can inject monoclonal antibodies into your bloodstream to help fight it. But this would not help to form any memory cells or anything like that. This is just like you're bringing in soldiers to get rid of an enemy, and then, the soldiers go away. So vaccines provide artificially acquired active immunity. It stimulates the production of antibodies due to intentional introduction of an antigen. They are usually multi-dose to simulate both the primary and secondary immune responses, and they are used for communicable diseases such as measles, HPV, and rubella. You wouldn't, for example, have a vaccine for diabetes or coronary heart disease because these are not caused by antigens or pathogens. So vaccines have numerous modes of action. This includes live attenuated vaccines, which contain weakened forms of the pathogens. We have inactivated vaccines, which contains pathogens that cannot reproduce, subunit vaccines, which contains a detectable component or protein of a pathogen, well, basically just the antigen, and mRNA or messenger RNA vaccines, which contain the mRNA blueprint of the pathogen component, so that your body can use that blueprint to build the little spike protein or the virus. For example, the Pfizer vaccine is like this, so it can be synthesized in the body for detection. So there are always certain advantages and disadvantages of using vaccines. So advantages are that the vaccines are often successful at stimulating memory cell production. So it often reduces severity of infection. Usually if you used to get reinfected, you might even know the virus or the bacteria might be defeated right away. The detection rate could be very high and the eradication rate could be very high. So this reduces incident rate. You have less people who have the disease. So less people will get potentially infected and spread the pathogen to the uninfected or the vulnerable. If you have enough of this generates something called herd immunity. Disadvantages of using vaccines, however, is that viruses mutate often. So remember, some vaccines only use a certain version of the virus or a certain version of the antigen. So viruses may produce variants. So remember Omicron and Delta and all these things for COVID-19. So the variants may be different from the versions that are found in vaccines. The vaccine may be ineffective against these variants. Next, vaccines may cause severe allergic reactions, though these are rare. And lastly, the immunocompromised may be at risk of getting ill with the vaccine. So if you used to use, for example, subunit, or you just use the antigen versions of it, you never know if this could affect someone who is very, very ill. Obesity is defined as having a BMI of over 30. BMI or body mass index is calculated by having a person's mass in kilograms be divided by their height in meters squared. Now, BMI is not the best index for calculating health. In this case here, we could have a bodybuilder who could have a lot of muscle mass and they might be accidentally classed as obese. But obesity typically occurs due to overconsumption of foods rich in carbohydrates and energy-dense lipids. And when consumed in excess, the molecules are converted for storage and become deposited in adipose tissues. These are fatty tissues, which can be deposited below the dermis, that is called subcutaneous fat, and around organs, that is called visceral fat. So this increases the chances of type 2 diabetes, cardiovascular diseases, kidney failure, and fatty liver disease. I say especially if it was to be visceral fat, which is deposited in or around the organs. So diabetes type 1 and type 2. Well, if you look at the diagram here, we see that the typical thing here is that the pancreas is going to release insulin, and that's going to allow glucose to enter the cells. The insulin is going to bind to insulin receptors on the cells, glucose is going to enter the cells. So type 1 diabetes, this is when the pancreas produces little to no insulin. This form of diabetes is genetic and is usually diagnosed in childhood. So patients will need to use insulin injections for all their life. Type 2 diabetes, on the other hand, is where the pancreas will still produce insulin, but the insulin receptors on the cells do not respond. This is called insulin resistance. And this is usually due to impairment of the cells or tissues that are caused by fatty accumulation and sedentary lifestyles. So patients usually must exercise frequently or take oral medication like metformin to help with the processing of the insulin. Diabetes type 1 and type 2. Well, if you look at the graph here, the y-axis shows blood glucose concentration in milligrams per deciliter, and the x-axis shows hours after eating a meal. Let's imagine that the person ate a meal here. So diabetes, whether it be type 1 or type 2, restricts the movement of glucose from the blood capillaries into the cell cytoplasm. This is going to result in fatigue and dehydration, and in worst case, tissue necrosis, that's tissue death, and amputation. In diabetes, assimilation or the uptake of nutrients by cells, particularly glucose, is delayed, especially when a large meal of sugars is consumed. So if you look at the person who doesn't have diabetes, there's the person in red. We can see here that they ate the meal, they've absorbed the glucose into their bloodstream, and now the glucose is being assimilated into their cells. This is because insulin is binding to the cells and is basically allowing the entry of glucose from the bloodstream into the cells. This is why the blood glucose level is going to drop. Now, with the person with diabetes, you'll notice that their blood glucose level set point is higher than the person who doesn't have diabetes. Usually, typically, if you're above 130 milligrams per deciliter at your fasting rate, this is like if you didn't eat anything, you have diabetes. So what's happening here is that when the person eats the same meal, or even larger meal, let's say, their blood glucose level is going to rise, and it's going to take a longer time for the glucose to assimilate into the cells. This could be either due to the cells not responding to the insulin, that'll be type 2 diabetics, or very little insulin being produced at all, that'll be type 1 diabetics. So factors that affect the regional distribution of diabetes, especially diabetes type 2, include 1. Diet and obesity. So the prevalence of fast food being the economic and convenient option, prioritization of foods high in lipids and refined carbohydrates. So for example, in Trinidad, we have plenty doubles here that there could lead to obesity and eventually diabetes. 2. We have sedentary lifestyles. So countries or areas with long hours of office work and no time for outdoor recreation raises risk of obesity and diabetes. Also, you have the advent of technology, which you have a lot more indoor than outdoor activities nowadays. 3. Prenatal malnutrition. So an ongoing study has linked malnourished mothers to children who are more likely to develop type 2 diabetes later on in life. Then we have age, ethnicity, and genetics. So of course the risk of diabetes type 2 increases with age. People of African and Asian descent are observed to more commonly develop type 2 diabetes. So let's look at atherosclerosis. So arteries have thick muscular walls and small lumens that facilitate high pressure flow. Atherosclerosis now refers to the condition where plaques form and adhere to the arterial walls, and this will thus reduce the lumen size. Remember, the lumen is the passageway of the blood, and this would limit blood flow. So what these plaques are made up of are deposits of cholesterols and chemicals from tobacco smoke. High pressure blood flow can transform the plaque. This is like if blood flows into the the plaque, this would transform it into a thrombus or blood clot. So if the thrombus was to fracture and enter a coronary artery, these are the arteries that surround the heart and supply the heart with oxygen and glucose. If the thrombus was to block these, the heart would not receive these materials, and this could eventually lead to a heart attack and potential death. Now, the decrease in lumen size means that less area is available for blood flow unless the heart was to apply more force. This would lead to elevated blood pressure or hypertension, and this is when the blood pressure is above 140/90 millimeters mercury. This refers to the systolic pressure. The 140 and the 90 refers to diastolic pressure. Now, stroke could also occur if the blood clot was to get deposited into a diseased carotid artery and block oxygen flow to the brain. So exercise is defined as activity requiring physical effort carried out to sustain health and fitness. So some short-term effects of exercise. So gaseous exchange is of course an increase in breathing rate and tidal volume, which is the volume of air circulating in the lungs. It also increases diaphragm contractions. This is caused due to an increase in acidity due to the buildup of carbon dioxide levels. These are detected by chemoreceptors in the arteries. Circulation is also affected, so this includes an increase in heart rate due to adrenaline secretion and increased activity in the sinoatrial node. Secretion of nitric oxide from blood vessels, these will widen or dilate the arterioles that would increase blood flow to the heart and muscles so that more oxygen can reach those muscles there. Also, the dilation of these arterioles, which is also called vasodilation, would lead to heat loss through the skin. This is why when you exercise and you put your hand over your skin and you feel some heat radiating out of it. So let's look at some long-term effects of exercise. So in terms of cardiac efficiency, long-term exercise increases red blood cell count and thus oxygen carrying capacity. It increases the heart muscle size and the stroke volume. More blood can be pumped out per stroke, and increases the maximum rate of oxygen use by muscles. This is also called the VO2 max value. In terms of long-term effects of exercise on muscles, this could increase the number and size of mitochondria in the muscle tissue, which would of course raise the rate of respiration. It could also increase the size of the cross-sectional area of the muscles and how much glycogen is able to store. It could also increase the number of capillaries in the muscle tissue, thus improving oxygen supply. All of this would help with strength and stamina, of course. Finally, we have body mass. Long-term effects of exercise would help to reduce body mass, or I should say like fatty mass in the body, which would reduce risk of chronic non-communicable diseases such as hypertension, type 2 diabetes, and coronary heart disease. Viruses and vectors. A virus is a disease-causing agent that can be transmitted from one organism to another via droplet spread, direct contact of vectors. Vectors would be like mosquitoes, flies, cockroaches. So example would be the dengue virus pathogen can be picked up by the Aedes aegypti mosquito, and this would be particularly female Aedes aegypti, from infected humans. So in this case here, the mosquito is the vector for the disease and transmit the virus to the uninfected. Viruses are not alive, they are non-living, and can only be eradicated by the immune system or injected antibodies. They cannot use antibiotics against viruses. If we look at the structure of a virus here, we can see that within there's a nucleic acid genome. This would contain RNA, and this would help to synthesize new viruses. We have little spike proteins, which are attachment sites to enter the human cells. Think of these as keys. We have the protein capsid that's going to help retain the RNA. We also have reverse transcriptase, and if you remember from Unit 1, this enables reverse transcription. Basically, it could help to turn this RNA into a DNA molecule. Lastly, we have the phospholipid envelope, which is the structure taken from a human cell membrane. Dengue fever is transmitted by female adult Aedes aegypti mosquitoes. So what happens here is that the mosquito injects its proboscis, or its mouth parts, into the skin and draws blood from the vessel. It would also secrete saliva, so the saliva enables this to happen without clotting. The saliva would actually have the pathogens in it. So if the bitten person has the dengue virus in their skin cells, the mosquito will draw it in as well and become a vector. So if this person has the dengue virus, the mosquito is going to draw it in as going to rest in his abdomen, the viruses. So if the mosquito then bites an uninfected person, the virus will enter their capillaries via the saliva. So how does viral infection and spread go? So first we have attachment. So attachment is when viral spike. So how does viral infection and spread occur? Well, first we have attachment. Viral spike proteins bind receptors on the cell membrane. Remember I said these are like keys. 2 is going to be entry. The virus is going to be taken in by a vesicle through the evagination in the cell membrane. This is endocytosis. The protein capsid is going to break down and is going to release the RNA into the cytoplasm of the cell. So 3 now is going to be replication and gene expression. So you're going to have the enzyme reverse transcriptase, and that's going to enable the RNA to make multiple complementary DNA copies of itself. Now this RNA and this DNA now is going to assemble into proteins. Basically, remember from unit 1, RNA is read by ribosomes along the rough endoplasmic reticulum, and amino acids attach in response to reading that in that set of instructions, RNA instructions, and that's going to build a protein. So these new viral proteins are synthesized within the cell itself. So the cell becomes like a photocopying machine for viruses. 5. The new viruses that are built are packaged into vesicles by the Golgi body and are released via exocytosis. And these viruses are now off to infect a new cell. So AIDS, which stands for Acquired Immune Deficiency Syndrome, is the most advanced stage of HIV infection. HIV invades helper T lymphocytes, which are called CD4 cells. The T cells' nucleic genetic code is rewritten to produce more HIV copies. This is basically the same thing as seeing here with dengue, where you had the viral penetration, reverse transcription, making new DNA copies, entry into the nucleus, integrating within the DNA in the nucleus, transcription, which is basically forming more mRNA molecules, and translation, which is assembling proteins which will then migrate to the cell membrane, bud off, and then be sent off to infect more white blood cells in this case. So due to the highly compromised immune system, because a lot of them being affected are the lymphocytes, the T lymphocytes, opportunistic infections may become deadly. Remember, these helper T lymphocytes help with both types of immune responses. They can signal to T killer cells and B lymphocytes to either produce antibodies or destroy any infected cells. Because these helper T lymphocytes numbers get reduced, a lot of other infections end up popping up, and that is what an AIDS patient may die of. Not of AIDS itself, but opportunistic infections. So symptoms of AIDS would be persistent fevers, lesions called Kaposi, sarcomas, and severe weight loss or muscle wasting. So HIV can be transmitted via the following. 1. Unprotected sexual intercourse due to exposure to semen, vaginal fluids, and blood if the anal or vaginal linings are damaged. It could occur through the placenta from the mother to the fetus or via childbirth, through breast milk from HIV-infected mothers to newborn babies, blood transfusions due to transfer of contaminated blood during surgeries or while sharing hypodermic needles, for example, if you're to be taking drugs. HIV can be detected by doing blood tests or by doing T lymphocyte cell counts. So when the helper T cell count falls below 200 cells per millimeter cube of blood, the patient is said to have AIDS. The incubation period for HIV is about 3 months, so regular screening must be done. And antiretroviral drugs, for example AZT, can be used to preserve T cell count, but they are expensive and not really affordable to the common citizen. So what are some factors that affect the regional distribution of AIDS? Well, one would be the culture. So some cultures discourage condoms and some encourage multiple partners. So stigma and unwillingness to admit HIV status or get tested, of course, that there could lead to a lot of infected people who don't know they have the infection to infect more people. We also have long incubation periods. So since HIV may not be detected by blood tests until 3 months into incubation, an HIV positive person can be tested in January and wouldn't know until March or April, and they can unknowingly spread it. Ease of travel. So whenever there's a large number of tourists, especially those partaking in sex tourism with locals, the virus can quickly spread to local populations. We have poverty and lack of education. So poverty would encourage sex work, and this facilitates spread if done irresponsibly. And there's lack of antiretroviral health care in some countries. So for example, if there's a woman with AIDS who is pregnant, their baby doesn't necessarily have to have AIDS. There's therapy that could help with that. And lack of educational programs that inform about HIV transmission and prevention. Cancer. So cancer refers to a group of diseases where cells in a certain organ or tissue uncontrollably divide. And age influences cancer risk, as it is usually formed due to a random mutation, usually during DNA replication. So carcinogens are also capable of causing cancer. This could be cigarette smoke chemicals, ionizing radiation such as X-rays or gamma rays, food additives such as sweeteners as saccharin, and viral RNA such as the one produced by the human papillomavirus, which causes cervical cancer. So factors that affect regional distribution of cancer would include the following. Well, one would be environmental hazards. Chemicals from factories or buildings such as asbestos cause cancer. Outdoor environments are subjected to frequent exposure to ultraviolet radiation from the sun. This is why you have SPF factors in sunscreen. Ionizing radiation from labs can also cause cancer if precautions aren't taken. 2. Viruses. So HPV, which causes cervical cancer, can be transmitted via sexual intercourse and may be more prevalent in regions without proper condom use or access to HPV vaccines. 3. Genetic factors. So alleles that express risk of developing certain cancers can be higher in concentration in certain countries. So for example, breast cancer is especially prevalent in the Caribbean. 4. Lack of medical access. So lack of mammograms which detect breast cancer or access to biopsies mean that it may be too late for some cancer patients to seek treatment. Typically, if a person knows that they have cancer very early on, the likelihood of survival is much higher. And the overall impact of diseases on society. Well, first, the economy. The strain on state-funded medical care if there's a lot of sick people around. There's the money for awareness campaigns and medical personal protective equipment. There's a reduced labor force, especially if a lot of young people become pregnant, and an overall drop in productivity. People can't work. Businesses are affected by lockdowns. We would have experienced that firsthand with COVID-19. 2. Trade relations. There's reduced income from exports, and tourism could be affected by stigma. Not many tourists want to go where they know they could get malaria and dengue easy. 3. Food availability. Food shortages can occur if the agricultural sector is impacted. 4. Household income. Privatized healthcare and medical bills, very expensive. You could also have unemployment if the breadwinner of the family is affected by a disease. The quality of life, which represents the emotional and psychological tolls of the illness and quarantines. And 6. Positive awareness. So sometimes when there's a lot of disease in society, this may influence certain individuals in society without the disease to make lifestyle changes and take extra precautions, such as washing hands more often or using protection when having sex. This is why the number of HIV cases would have dropped over time. Prevention and control of viral infections. Well, first we have social factors such as quarantine and safe sex. Well, quarantine limits the spread of viral infection, but may have a severe effect on businesses and household earnings if it was to result in lockdowns. Safe sex ensures the use of condoms and limiting the number of sexual partners, and this would prevent sexually transmitted infection spread. Biological factors would include PPE, such as surgical masks to prevent droplet spread, and sterilizing medical equipment. Vaccination reduces the incidence of disease and hence the spread of infection. Good hygiene, such as regular hand washing, use of disinfectants and antibacterial soaps, also reduce the incidence of disease and spread of disease. Regular blood screening limits the spread of pathogens such as HIV, and antiviral drugs and monoclonal antibodies can be used to treat some viral infections and thus reduce the spread. Economic factors will include pest control, awareness campaigns, and the cost of preventative medication. So pest control would be like for mosquitoes, for example, removing stagnant water sources and keeping refuse from piling up. This would reduce the chances of any vector breeding, whether it's flies or cockroaches or mosquitoes or rats. Insecticide spraying programs, introducing fish to ponds can eradicate mosquito larvae and eggs. You also have mosquito nets and coils. Awareness campaigns, well, education about limiting spread and infection always helps. And cost of preventative medication, surgical masks, PPE, private doctor visits, blood screening, mammograms, biopsies, all of these here cost money. Every factor here costs money, but they can help to limit and control viral spread. Drug abuse. Well, a drug is an externally administered substance that alters the body's physiology. Drug abuse occurs when the use of this drug leads to physiological or psychological harm to the user, their family, or society at large. So remember, a drug, there could be the good prescribed drugs such as penicillin, but you could have the bad ones such as cocaine and heroin. So drug abuse leads to drug dependency, withdrawal symptoms when the drug is not used, and possibly overdose if too much of the drug is used. The progressive decrease in response to a drug is called tolerance. So for example, if you was to take heroin one day, it'd feel real good, but then the next day you'd have to take a little more to have the same response to it. So there are 4 categories of drugs. Stimulants, which include nicotine, cocaine, and caffeine. These basically stimulates the nervous system. Depressants, examples would be alcohol and codeine. This basically delays activity in the nervous system. Hallucinogens, which basically amplify emotions and cause hallucinations. This includes cannabis, LSD, and PCP. And we have opioids, which act as analgesics or painkillers. This would be like heroin, morphine, and fentanyl. So there are 2 types of drug dependency. There's psychological dependency and physical dependency. So first we're going to look at psychological. Let's look at a case study here. Wayne is 16 years old and smokes cannabis with his friends. He often goes without cannabis for days at a time with no ill effects. However, after a week has passed, he feels a strong urge to smoke, especially when he feels depressed or bored. Well, Wayne is showing psychological dependence, and this is characterized by lack of strong withdrawal symptoms because he could basically go for days at a time with no ill effects. He has the ability to go prolonged periods without the drug, as just said, and drug abuse usually occurs due to social influence or boredom. So let's look at this other case study. Sandra is 40 years old and uses heroin in 2 to 4 injections 4 to 12 hours apart. When she goes longer than 12 hours without heroin, she feels sick and becomes anxious. In this case here, she's showing physical dependence. This is characterized by very strong cravings, strong withdrawal symptoms if the drug is abruptly stopped or reduced for short periods of time, and the symptoms would worsen as the frequency of drug abuse increases. So there are many health consequences of alcohol abuse. One is impaired nervous transmission. So since neurotransmitter activity slows down, reaction time decreases. This is why you shouldn't drink and drive. And perception becomes distorted. For example, blurry vision, reduced motor skills. Due to the cerebrum and the cerebellum, those are parts of your brain that deal with memory, judgment, and coordination being affected. Dehydration. So the pituitary gland is also affected by alcohol. So ADH release is suppressed. If you remember here, ADH is used to put aquaporins on the collecting duct of the nephrons. This would help with the reabsorption of water. So this means that less water is reabsorbed into the blood from the kidneys, and this could lead to dehydration and hangovers. There's an increased risk of cardiovascular diseases because alcohol allows calcium ions to bind to blood vessels. This will cause them to constrict. This increases the chances of a blood clot rupture and an ensuing heart attack or stroke. So what happens during liver cirrhosis? Cirrhosis means scarring. So alcohol is usually broken down in liver cells or hepatocytes by an enzyme called ethanol dehydrogenase. This would convert alcohol to ethanoate, which would then eventually enter the Krebs cycle to produce ATP. That's what you're seeing here. Ethanol to ethanol to ethanoate. So during this reaction, NAD is being reduced to form NADH. So remember, NAD would basically catch these hydrogens that this hydrogenase would be letting out. Now the problem here is that the liver is using this NADs to break down ethanol. But NAD is usually used in the liver to oxidize fatty acids. So you don't have enough NADs now needed to oxidize and break down these fatty acids. So these fatty acids begin to accumulate in the liver due to the decreased availability of NAD. These are stored as lipids in the liver cells, and this is going to result in fatty liver disease and scarring or cirrhosis. So let's look at some components of cigarettes. So cigarettes has tar and particulates, and these lead to irritation in the airways and the breakdown of the protein elastin. This is going to create large air spaces in the air sacs or the alveoli. This would reduce the surface area for gaseous exchange. This is called emphysema. Next, we could have excess mucus production, basically is an irritant, and this would provide a breeding ground for bacteria. Ciliated cells, which usually trap dust and bacteria, are going to be destroyed. So this could increase the risk of bronchitis bacteria entering the airways and the blood. Carbon monoxide. So remember, hemoglobin has a very high affinity to carbon monoxide. This means that carbon monoxide is going to bind very readily or quickly to red blood cells instead of oxygen. And because the body now is going to be deprived of oxygen, this could lead to potential asphyxiation and death. In fact, inhalation of carbon monoxide is a popular suicide method. Even being in an environment of smokers and inhaling the components poses a threat. This is called secondhand smoke or passive smoking. Now remember, cigarette smoke also contains carcinogens, which are cancer-causing agents as well, such as for lung cancer or throat cancer. Lastly, we have the effects of nicotine. First of all, nicotine is a stimulant. It closely resembles acetylcholine and tends to bind to acetylcholine receptors in synapses, the postsynaptic membranes. However, since acetylcholine esterase cannot break down nicotine, it stays bound for prolonged periods of time. This will cause the synapse to be overly excitatory. So you could think of it as kind of like a competitive inhibitor for an enzyme. Nicotine constricts blood vessels. In arteries where atherosclerotic plaques have built up, this greatly increases the possibility of a ruptured blood clot. The effect of nicotine on pregnant women's fetuses can cause much prenatal harm. Nicotine causes the constriction in the placental blood vessels. This would reduce the exchange of nutrients and gases between the mother and the fetus. Nicotine is highly addictive. People who regularly consume nicotine usually exhibit physical dependency on it. So when suddenly stopped or reduced, it can produce a myriad of withdrawal symptoms. This may include anxiety, depression, cravings, and irritability. The nicotine from tobacco relaxes the valve between the esophagus and the stomach. This is the lower esophageal sphincter. This can allow stomach acid and juices to build up there. This would result in reflux into the esophagus. The chemicals that break down food in the stomach would reflux, and this would cause heartburn, peptic ulcers, or stomach ulcers, and nausea. Lastly, nicotine changes chemical processes in the cells to increase insulin resistance. Remember, insulin resistance is a precursor to type 2 diabetes, and this basically delays assimilation of glucose from the bloodstream into the cells. This will result in a number of issues, including dehydration, nerve damage, cell death, and coma. And that is Escape Unit 2 Biology. That's it. Good luck. Have a good day.